problem. There are many other problems, and microbiology is the only tool which can reduce the many problems in India and help the government. Always I'm telling to my student, you are studying microbiology to get the job. Once you get the job, what is next? Next is serving the society, serving the nation. So we can serve the nation by using microbiology subject. So one more example I would like to tell again. Genetically modified food, GMF. The nearest country, Bangladesh, has allowed genetically modified brinjal. I was in USA. I was in European countries. I have seen that in all these countries, there are genetically modified apples, there are genetically modified grains, there is a golden rice and whatnot. Many genetically modified foods are available. Unfortunately, in India, there is only BT cotton. No other food, no other crop. Neither earlier central government nor present central government is giving permission to the genetically modified crops. Now it's the duty of you and me to create a pressure on the government, and we should force the government to give the permission for BT brinjal, BT apple, and there are many other BT crops are waiting. So that duty we can perform for the development of nation. Again, you see, the quality means, again, I'm telling to my students, and today many students are attending this webinar. I'm telling to my student that microbiology is the subject where sky is the limit. Sky is the limit, meaning is that for every problem, microbiologists are required now. I will give the example. Our Honorable Prime Minister have declared that we are very, very proud because two of the companies producing vaccine, two best companies producing vaccines are Indians. That is a Covishield and Covaxin. And which scientists are involved in production of vaccine? Let me tell you proudly, all are microbiologists. I'm from Pune. In Pune, there is a Serum Institute of India. And more than 100 my microbiology students are every day working in Serum Institute of India. In all pharma company, who is working and who is producing tablet capsules and the vitamins and tonics? Only microbiologists, they are involved in the quality control. You see, quality control, where the quality control is required, anything which we are eating, any food material, any drinks, anything which we are eating, anything which we are drinking, anything which we are engulfing. Engulfing meaning is that we are engulfing tablets and capsule. So anything which we are engulfing, anything which is injected in our body through syringe and needle, and anything which we apply on our body surface, right from powder, right from lipstick, right from nail paint, right from spray, and many, many other things. Everywhere quality control is required. One time, when I was a professor, many industrialists came to my department and requested me, sir, we want any of your student for a job in my company. I told, which company? He told, all they were telling, we are producing pure water, Bisleri water, and government now has closed our industry and asking that without microbiologists, you cannot start your water purification industry. Meaning is that Everywhere now the microbiologist is required. Even in the today's situation, the COVID-19, if you go on internet and search with my name, you will find that I have made many videos to fight with the coronavirus, to educate the people, to create the awareness. And I was requesting my student, to all microbiology students, you are the ambassadors. You are studying microbiology. You know what is microorganism. You know what is antibiotic. You know what is vaccine. Educate the people. And I'm sure now, with your infrastructure, with your achievements, with your standard of your college, definitely I will be happy to promote you more and to bring you at an international level. And you see, all neighboring countries are looking toward India. They are waiting that India should lead and guide to us. So we can do it definitely. I'm very sure. And that's why I'm very happy today to inaugurate the student unit. I take an opportunity to tell the ma'am that today's student unit is inaugurated and will be functional from today. And you carry on many activities. Microbiology Society is very happy. We are always in search of active persons. Your Vandana Sharma ma'am and Sandeep Kaur ma'am both are so active that I am very happy to support them, not only morally but financially also. We will support them, and for the betterment of India and to create the good fate of the students, we should work day and night. So I will request the student that you are a young, you are a dynamic. Don't underestimate yourself. See, the greatest drawback in the development is that generally we underestimate ourselves. You are thinking that we are BSc students, undergraduate students. Sir, I'm only undergraduate students. What can I do? You can do many things. If you decide, if you are having ambitions, two things are required: ambitions and guidance. So, guidance will tell you on which way you have to proceed. What are the fields in the microbiology and biotechnology? And you see, there are many fields there. India have a history, very good history. One name I would like to tell again: Ananda Chakravarti. Ananda Chakravarti was a great scientist, died 6 months back. He was from India. You go on internet, you search in the book, you will find the name of Dr. Ananda Chakravarti, who first time produced genetically modified Pseudomonas aeruginosa. He called it as a superbug, and it was the revolutionary innovations in the world. And like Dr. Anand Chakravarti, there are many other scientists. So again, I will request, be dynamic, be active throughout the year. Now 3 or 4 lectures are there today. Listen them carefully. They are all studied, and they are going to talk on the how to control the COVID-19 even in the future, and other contagious diseases even in the future. And believe that in the future, world will be ruled by microbiologists only. You tell me any problem. Again, I am telling you, you take any word and a suffix bio to it, it's a biotechnology. You take a biocement, whether you know or don't know, I don't know. Biocement is there. One of my PhD students worked in UK, in Bath University, on biocement. Biosteel, biosolubilization, that is, sorry, bio-leaching, bio-bleaching, bioterrorism, bioremediation, biostimulation, biofertilizer, biopesticide, biobatteries, and I can tell a list of more than 100 words where there is a bio is a suffix. Meaning is that for every technology now, all administrators, all presidents, all prime ministers of the world, they are telling that use bio, don't use purely chemical, purely physical weapons. Come to the bio. So we have to, means we are ambassadors. It is my duty. It is your duty to educate the people, to help the government, and to serve the nation. And our microbiology society is serving the nation. Our goal is to serve the nation and ultimately to serve the mankind. And I'm very happy. I'm from Pune and all you are from Chandigarh. Distance doesn't matter for good work. Whenever in your mind you want to do something good, you are approached to me. You have started a student unit, and that is the beauty of virtual lecture. I can speak from Pune. You can hear from Chandigarh. Many other participants may be there from different corners of India. So, again, ma'am, really, I must be thankful to you because it is my goal in the India, and you are ahead than my goal. You are doing a very best thing, and I am sure in the future, under the umbrella of your Bhargava ma'am, definitely you will do achieve many things so that your college will be proud and your city will be proud of you, your colleagues will be proud of you. So thank you very much again. I am very thankful to you, and I give all the best wishes for today's international conference organized by your college in collaboration with the Microbiology Society. Again, at any time, I will come on your platform as per your wish. Because always I am very happy to interact with the student. We will arrange a Google Meet with the student so that students can ask question, interact with me. But we'll do many good things, many better things. And as a president of Microbiology Society, it's my duty to wish you all the best. Thank you, ma'am. Thank you. Thank you again. Thanks very much.
Thank you so much, sir, for the informative talk and for the kind words. And we really look forward for more future collaborations with the Microbiologist Society. And thank you for enlightening our budding scientists about the role and scope of microbiology. Yes, microbiology is the in thing, and we are proud that we all are microbiologists. Thank you so much, sir. And to take out your time from your busy schedule, we highly appreciate it, sir. Vanna ma'am, would like to say anything?
Thank you so much, sir, for your blessings and your best wishes. So we'll definitely will try to do more and more activities in collaboration with the Microbiology Society. So thank you, sir, for sparing your valuable time and joining.
Ma'am, students have any questions?
Yeah.
Related to
Yes, they can ask the question.
Students,
student want to.
Yeah, students, you are free to use this platform and interact directly with the expert. Please, students, do ask your questions. Any questions related to microbiology or COVID-19, sir is here and it's a golden opportunity. Don't miss on this. Yes?
Sir, I have a question.
Yes, one student is there.
So
Yes,
since
yes.
COVID-19 is mutating and is becoming more infectious, like the UK strain, and there are rumors also that US strain is also coming up. So what if the vaccine is not effective against the coming up strains? Then what should be the next possible step we should be taking in order to curb the infection?
Yes, but for your answer, at least 1 hour I may take, but I will not take it now. Because you see, this new strain is coming, UK strain is coming. And it is slightly mutated. I'm using word slightly. So Indians should not be much worry about that strain. There is only one change on one epitope. It has increased its infective ability, spreading ability. But the same vaccine is useful for that strain. And again, one more thing I would like to tell you. We Indians have best immunity as compared with the Western people. That's why the cases are increasing in India. And why we Indians? The reason is that we are having more habit to walk. You see, not like Western countries. They travel only on the four-wheeler, sit in the AC. But we Indians are having habit of walk. We Indians go in the sunlight. Western people, they never see the sunlight for a month together. We eat simple water. We drink simple water. They drink only Bisleri water. So our immunity is a very best as compared with the immunity, or not only our, in SARC countries, you see the same situation in Pakistan, Bangladesh, Nepal, Bhutan, Sri Lanka. In Sri Lanka, one ma'am, biotech ma'am is there. She told, sir, in Sri Lanka, it is as good as there is no coronavirus. So the situation, we Indians should not worry about the new strain. And our present vaccine will definitely fight with the new strain. Again, sometime later on, I will talk 1 hour only on that immunity and mutations in coronaviruses. Thank you.
Sir, I want to ask about herd immunity. Sir, does it work?
Surely today in India, cases are decreasing. Why? Only because of the herd immunity. And you see the sero surveillance. Sero means serum. Surveillance of serum, survey of serum shows that antibodies are present in 50 to 55% people blood in slum area. In rich area, it is only 10 to 15%. But herd immunity works. Only the disadvantage of the herd immunity is that we have to give the cost. Which cost? 1% death. That's why in the whole world, herd immunity concept was not accepted. Vaccine concept is accepted. Okay. Anything?
Okay, sir. Thank you, sir.
So I
So
have a
yeah,
question.
Deepika is asking. Who is asking?
yes, so I'm asking. So there's a question that it's by Sahaj Kakkar that now we have the vaccine. What if there are some complications? Not now, but for the future.
You see, even you take a polio vaccine, BCG vaccine, all vaccines, you go in the book, find out the literature. There is a possibility of complications, 1 in 10 lakh, correct? And mild complications, almost in every child, there are mild complications. So the chances are there. But whenever 9,99,999%, if we can save the life. Only one person, if died because of the vaccine, the science allows it. So there are chances, no doubt, but very, very minor chances are there. Least chances are there. One who is having very, very weak immunity, he may suffer, other will never suffer. So there are chances. Then one more, Seva was asking. One more question was from Seva.
सर यू वर टॉकिंग अबाउट फर्टिलाइजर्स सर कुछ टाइम पहले फिर फर्टिलाइजर इंट्रोड्यूस क्यों हुए थे बिकॉज किसी चीज को इंट्रोड्यूस करने से पहले बहुत टेस्टिंग होती है नाउ वी आर सेइंग कि कैंसर इज कमिंग अप सो उस टाइम पे क्यों स्टॉपेज नहीं लगा एंड व्हाई they are taking this issue now?
Very, very good question. I will tell you the story. Endosulfan was a chemical pesticide. It was used in India. WHO was writing to India, don't use endosulfan. It is a carcinogenic. But Indian Prime Minister was telling that we will endosulfan. Now your question. Although we were knowing that endosulfan is a carcinogenic, why we were using endosulfan? The reason was very simple. Our Prime Minister at that time told that if we use endosulfan, 1,000 patients will die because of the cancer. If we don't use endosulfan, there will be scarcity of food and few lakhs person will die because of the food. It is, in the statistic, it is called as a type 1 error and type 2 error. So when one error is increased, other error will decrease. If we use endosulfan, per patient will die because of the cancer, but the crop production will be excess, no one will die because of the food. That's why government was reluctant. But in 2006, all other countries compelled India and now it is banned in India. So there are many reasons. Again, as I have told you, to talk on that, I will require 1 or 2 hours more. We'll talk sometime. You can request your ma'am, arrange my lecture at any time. मैं तो कभी भी राह ही देखता हूं कोई बोलता है क्या मुझे माइक्रोबायोलॉजी पे बोल के. My topic is only microbiology, no other. So we'll talk sometime again. Okay? Okay, Vandana ma'am, I think.
Yes, sir. Can you please guide our students like the good microbiology institutes where they can go for the post-graduation?
Yes. Very right question you have asked. Whenever I'm going, मैं चाइना जाता हूँ, अमेरिका जाता हूँ, UK, यूरोपियन कंट्रीज, जहां पे भी मैं जाता हूँ, my students always they are coming to airport. Why I'm telling to your student? That is, many students from India already have gone for post-graduation, PhD, postdoc in the Western countries. And I will tell you, many scholarships are waiting in biological sciences as compared with the physics and as compared with the chemistry. So, first, they have to take the pass with the examination, different examination for entering into USA, Singapore, Japan and other countries, score good marks in that. And there are many in India, you see. All ISER institutes are best in India. There are about 6 to 7 ISER institutes in India. You go on internet, Google पे जाओ, type ISER, IISER. They are the best institute in India. Then CCMB is there in Hyderabad, which is one of the best. All IITs are best for research. And again, I will tell you, you don't complain about the money. Scholarships are available. Study करो, अच्छे मार्क्स स्कोर करो. और दूसरा, guidance. As Vandana Sharma ma'am very rightly asked the question, कि बच्चों को तमन्ना तो होती है, बड़ा होना है, लेकिन कैसा होना ये मालूम नहीं होता. We are here, ma'am, to guide the students. तो Number of fellowships are there. We'll arrange one-time lecture on it. There are many good institutes in India and abroad. 4 दिन पहले हमने बांग्लादेश का रखा था Google Meet. That director told me I will take 1 or 2 students from India if they come for PhD. I will provide a scholarship, PhD scholarship also. So like that, many chances are there for the student. Okay.
Thank you so much, sir. So one last question from my side, personal, that if in case students want to join for the international training program, can the BSc final year or second year students also go for the international training if you allow, or only MSc students can go? What are the requirements?
Any student,
Okay, okay, any level.
No ban, no barrier.
Okay, okay.
Usme I will discuss more with you after the seminar within 1 week, within 1 month.
Yes,
Lekin
sir.
in Maharashtra, there was a one team from Nepal, one team from Bangladesh. They enjoyed in Baramati training and they wandered here, visited many institutes. Likewise, many teachers also visited Nepal. Many teachers, because suppose by chance I will tell you, ma'am, you are going in Nepal for a tour, for example. But you ring, you told me that is a, sir, I am going Nepal. Can I deliver one lecture in university? Yes, I will give you opportunity to you. From all these countries, in every university, there are my friends. So like that, will increase the communication and ultimately the development of the student. Okay?
Thank you, sir. Thank you very much for taking out your precious time and answering to all our queries, and we really appreciate you, sir. Thank you, sir.
Thank you, thank you, thank you.
Thank you so much, sir, for joining and taking your time from your busy schedule and gracing this occasion. So thanks a lot, sir.
Okay, okay. So I will leave now from this meeting and we'll again meet sometime and we'll have definitely more activities.
Yeah, sure, sir.
Okay?
Sure,
Sure,
sir.
sir.
Okay. Okay.
Thank you so
Sure,
much,
Thank
sir.
you.
Thank you so much,
Thank you,
sir.
sir. Thank you, sir.
Thank you so much, sir. Thank you.
Thank you.
Okay, so hope students, this was really very fruitful and very informative session for all of you. We have been time and again focusing on the role of microbiology and the emerging role of microbiology, especially in today's scenario. And sir has really, emphasized the fact that microbiology is gonna be the in thing for today and decades coming. So we thank sir for the enlightening talk, and we'll have with us the next speaker in a while. Kindly be patient. In 5 minutes, he'll be joining us.
So meantime, you can go and have some water or some tea. So there are 5 minutes. So he will join at 4 o'clock, right?
The attendance link has also been shared for the first
Yes,
session.
ma'am.
Please kindly mark your attendance. It's on the quill.
Yes, in the meanwhile,
It's on the YouTube also and in this also. Meet also. The link has been shared.
Sandeep?
yes, Gurpreet.
संदीप वो टेलीग्राम वगैरह दूसरे वाले उसमें आप मैसेज डाल दो because some people they are like trying to get admitted into this meet but there is upper limit 100 has already been reached so if they can be informed to join YouTube or FB.
Yes, yes, sure, Gurpreet, I'll do it.
Okay.
Deepika, just a reminder that when I'll be introducing our next speaker, then you can share that screen. That's it.
I'll do that. This is the one.
Deepika, I just saw the message. Have you circulated the feedback link to Preeti? She was just asking. Attendance and feedback, not the feedback, attendance link to Preeti for this session on YouTube, to be shared on YouTube.
Yes, ma'am. YouTube pe I've shared the link twice, and I'm sharing it
Thank
for the next time also.
Thank you. Thank you.
Yes, ma'am. Sure.
Deepika, please share that in the chat box also. यहाँ पे वाले students भी fill कर देंगे. Whenever you feel
यहाँ
like
भी
कि
कर
ये
दिया
वाले
मैम.
तो
हाँ हाँ.
okay, okay,
यहाँ पे भी
okay,
शेयर कर चुके हुए हैं. Yes, ma'am.
okay, okay, done.
Okay. दोबारा शेयर कर देती हूँ.
Yeah, all the students, please fill the attendance form.
Okay, ma'am.
So I think meantime we can click a pic. So all of you, if you can switch on your videos, then we can have a pic right now. Anji, students, please. Thank you. हो गया बच्चे. रिदिमा, आप भी पिक ले रहे हो? एक जना कोई स्टूडेंट कौन, किसकी ड्यूटी लगी हुई है?
पता नहीं मेरी तो
संदीप, रिधिमा है? रिधिमा,
हाँ जी मैम।
हाँ बच्चे, आप भी जब कैमरा ऐसे ऑन हो ना तो आप भी पिक ले लिया करो साथ में, ठीक है?
ठीक है
और
मैम।
मीन टाइम बीच में जो है वो लेते रहना पिक्स।
ओके मैम।
Just I think 2 more minutes. Sir is joining. Sir is joining in 2-3 minutes. Just 2-3 minutes.
ओके।
मेक श्योर जब फोटोज लो आप तो प्रेजेंटेशन मोड कंप्लीट स्क्रीन पे हो, ठीक है, जैसे अभी आपको विजिबल है।
Deepika have shared Preeti ma'am's number in the unofficial group, and she
Okay,
has
ma'am.
just messaged me that she needs the attendance link.
मैं पेज
So,
संदीप।
okay, okay, ठीक है।
ओके, मैंने भी अभी शेयर किया मैम आपके साथ ना द सेम लिंक।
संदीप,
हाँ जी मैम, बोलो।
प्रीति ਨੂੰ ਨਾ इंस्टाग्राम ग्रुप च ऐड कर लो प्लीज, ताकि कुछ भी टेक्निकल बच्चों ਨੂੰ इशू आ रहे हैं तो वो ਉਹਨੂੰ ਨਾਲ ਉਹਨੂੰ पता चलता रहेगा।
इंस्टाग्राम ग्रुप
मैम फेसबुक पे, इंस्टाग्राम फेसबुक,
सॉरी इंस्टा नहीं, वो टेलीग्राम ग्रुप।
टेलीग्राम।
मैम वो एक्चुअली टेलीग्राम ग्रुप ना तुसी बनाया है, वो तुसी ऐड कर सकते हैं।
ओके।
Good evening, everyone. Sandeep here.
Yes, yes, sir. Yes, sir.
Yeah, good evening,
Are
sir.
you able to, am I audible?
Yes, sir.
Good evening, sir.
Yeah, good evening. I'll wait. It's, only 4:05 now. So we need to start at 4:15. I'll wait for another 5 minutes, then I'll share my screen.
Okay, sir, you can start right now.
In the meantime, I will, ma'am, I'll introduce sir and then he can start. I think we can directly. Okay. Deepika, can you share the screen? Thank you, Deepika.
Welcome.
So, yes, students, let's move forward to the next session. Uh, the COVID-19 pandemic, as we all know, has made us realize the importance of drug discovery and the fact that the road to development of a new drug is not an easy one. Other factors that besides COVID-19, I would like to say that there are many other infectious diseases, lifestyle diseases, which are equally worrisome. And there are leading causes of deaths worldwide, either due to lack of an effective drug, or in many cases due to lack of enough money to buy the drugs, as faced by developing countries and the people, common people, including India. So here comes the role of another important era in healthcare called biosimilars. Biosimilars is an avenue that assures a consistent and affordable supply of equally efficacious drug called biosimilars to patients in times of need and at affordable cost, thus saving the lives of many. Now let's learn more on this aspect of drug development and how it is done directly from the expert in this field. I extend a warm welcome to our next enthusiastic and prolific speaker, Mr. Sandeep Pathak, Senior Director, Quality Control, Biocon Biologics. Let me have the honor to introduce you to our young audience, sir. Mr. Pathak hails from Chandigarh. He completed his graduation and post-graduation in microbiology honors from Punjab University, Chandigarh. Sir has a total of 18 years of enriching industrial experience. He has worked in reputed pharma and biotech companies of India such as Panisha Biotech, Intas Pharma, Lupin Pharma, and now Biocon. He specializes in quality control testing of biologics with 7.5 years in prophylactics, that is vaccines, and 10 plus years in therapeutics, microbial and mammalian based, and quality assurance functions. He's a leading expert in optimization of analytical, physicochemical methods, microbiological methods, raw material packaging, in vivo, in vitro testing, animal and cell-based, analytical method validation, stability management, data and audit trial management, risk management, management review board, product recall, employee training with complete knowledge of GMP and regulatory requirements. Well, the list is actually very long. I've taken, on a personal note, I want to share that he was my senior. He's a happy-go-lucky person with a positive bent of mind, and he's full of encouragement and enthusiasm. So here we have with us Mr. Sandeep Pathak, and I thank you, sir, for giving your consent for today's talk at a short notice. And we will appreciate if you could, besides the main topic, also tell us a bit about your industrial journey. So we welcome you, sir, once again. And now over to you, sir.
Thanks a lot. Thanks a lot, everyone. And thanks a lot, Dr. Sandeep, for giving such a big introduction. Really thankful for that. So as she has briefed, I joined industry. It's almost been 18 years, and I say still I am learning. Say in today's presentation also, even during interactions, say I believe we can learn from each and every person. Maybe a person who is a fresher, people who are from academic stream. If we have the correct attitude, we can always learn what the, we have always, if we have the learning attitude, we can learn anything and everything from what our routine life is offering. So if you permit, can you please unshare your screen so that I can move to my presentation?
Yes, so please go ahead.
Sure, sure. Just a moment, it is taking a little bit of time. Are you able to view my screen? Hello.
Sir, it came for a while, but it is gone.
Okay. I'll just project it again. I'll just share it again.
Okay, sir. Okay.
Are you able to view it now?
Yes, sir.
Yes, sir, it's visible now.
Okay, and then now I have put it to this presentation mode. Is it fine now?
Yes, sir, it's good.
Okay, so good evening, everyone. So I would like to start this presentation by just giving you a brief on the biosimilars. As Sandeep has already told, so I'll go into the technical details, what are biosimilars, but what led to the start of biosimilars. Say there are several treatments. If we take the simple example, a layman example of a mobile, say there are companies like Apple, and the latest technology, it is very costly and not possible for each and every person to afford it, right? And similarly, there are so many brands. We normally say there are mobiles available across from different manufacturers across the globe, rather not to name any specific country. So which whereby we can get the same configuration, same functions at a much cheaper price. Now the only difference is the mindset and the quality standards when it comes to those electronic gadgets is, we say that in terms of the robustness, they are not up to the mark, or they may be a substandard quality. But that is not the case in, that is not the case in biosimilars, since the manufacturer needs to prove that it is quality wise similar to the innovator product. Based upon that thing only, that assurance only, the regulators will audit you, they'll scrutinize your product, your data, and based upon that only, they'll give you a go ahead for the approval on the marketing authorization. Now, before I go into the discussion, so all my points which are cited during the presentation or as a part of discussion are totally my personal opinions, and Biocon or any of its affiliate company brands has no linkage to it. So whatever I'm giving an example is not supported or is not claimed by Biocon. This is my personal opinion, and what I am sharing is based upon the knowledge what I have gained over the years of this industrial experience. And secondly, considering the copyright issues, the data confidentiality issues, so for the sake of representation, I have taken images and I have taken pictorial representations from the public domain, and say my intention is not to infringe any of the copyright issues, and with due respect I have mentioned the source of each image or representation where I have cited it, just as a thanks gesture to the originator. So post this, I'll come to my topic. Now, since as I can understand, majority of the audience today is from the biotech stream only, and we are all versed with we know the basics, what biotech is. Now, the majority of the industry, few years back, maybe 20 years back, was majorly pharma only. And majority of the people are comfortable or they know much about the pharmaceuticals only. Since few people ask me, what do you do in your office? Say, if you want to explain it to a layman or someone in a village, so I say that we fill the powder in capsule so that it becomes a capsule. We get the loose caps, we crush the tablets, we put it into a capsule, and that is what we do in my company. Now, putting that on the lighter side, just to give you an idea what challenges are there when it comes to a biologics when compared to a pharma. So I have listed the basic, the main points which are different in the pharma and the biological products. So pharmaceuticals are chemically synthesized and they have very simple structure. Now, based upon the analytical techniques available on date, we can 100% characterize the chemical product that is a pharmaceutical product, and we can as such copy it. Say, if we know the structure, say, everyone might be aware of the generic medicines. So generic medicine is nothing but exact replica or a copycat of the innovator product. If a drug which is available for X price is an innovator drug, then a generic drug will be available at maybe 1/10 the cost, but it has the same, the activity or the active components are the same. Here the main thing is chemicals being, the pharmaceuticals being chemically synthesized are easy to replicate, and we can make one-on-one replica of a pharmaceutical product. Contrary to this, the biological products are produced by microbes or by organisms. Say, we take the example of E. coli based platform or a mammalian cells. So because of that, they have got a complex structure, and this structure itself is very difficult. I'll not say it's impossible to characterize, it's difficult to characterize, and depending upon the type of molecule, a battery of sets is required to characterize the structure. The further complication is how to replicate that structure to get the functional activity. Since the major scope of this uh, conference was the vaccines, so the only difference, if you already have gone through a presentation on vaccines, if you see in vaccine, the emphasis is not given so much on the structure. But if you see in case of therapeutics, structure is the main, because everything is targeted towards a particular marker. So we mean to say that everything is specific to a particular marker which we are targeting, right? And within biologicals, there are two types of molecules, the prophylactics and therapeutics. Prophylactic and therapeutics are general approach which does not apply only to biotech products, also to a pharma product as well. Say the moment we go to a dentist for a root canal, initially the dentist recommends that you take this antibiotic, a mild antibiotic, for 5 days, although at that stage you're not having any infection. But that antibiotic is given as a prophylactic, so that once you undergo that particular thing, the chances of infection decrease. And once you have the invasive surgery, then post that you're given a comparatively stronger antibiotic. And I always believe in telling the other way around, so it's easier to remember. The beating what the kids get from their parents prior to exams, that is a prophylactic beating. They sort of beat you to ensure that you study for longer hours and you get good marks. But once you do not achieve good marks, that beating post your results, it becomes a therapeutic. So a same medicine can be used as a prophylactic as well as a therapeutic. Now this slide just gives you a diagrammatic example, just illustration on a pharma versus a biotech product, that is a mAb. Say this structure on the right side, which is the ring and the chain structure, it is of the aspirin, and the other one is of a mAb. So here, if you see in case of a aspirin, paracetamol, it is a simple structure which can be 100% replicated to give a product which is, you can say, similar to the innovator product. But if you see the example of monoclonal antibody, here I would like to put you for easy understanding. You say that your protein, say if a female having long hair leaves her hair straight, so that single hair straight is a protein. It's the primary structure. Then you take the hair and you put it into a braid, choti or gut. I'll use sometimes the vernacular words for easy understanding. So if you make a choti, 3 chotis out of it, so that is the secondary structure. And then if you merge these 3 chotis into 3 judas, that will become a tertiary structure, and those three Judas, if you merge together, it forms a quaternary structure. And for the sake of beautification, if you put some clips, some pins, some, you may say, beads, so that is something which is the post-translational modification. So this is what happens in case of the monoclonal antibodies. So it's not only the primary structure, it's the way the protein folds. If your folding is not precise and accurate, so it will not give the biological activity. Now, coming to what are biosimilars. So as I emphasized in my first slide, that the generics are exact copycats, the replica of the innovator products, but the biosimilars have a special requirement. I have highlighted the parts. The active substance must be similar to the one in the reference biologic. It means, say, you might be aware of the word patent, and the innovator products are patented for 20 years because the innovator has spent so much money and so much time on the research and development of that molecule. That is why they have got the advantage. Now, once the patent goes off, the biosimilar can be manufactured. If we find that there is a difference in the excipient, we can change the excipient, or we can change any ancillary thing which does not impact my biological activity, my clinical effect. Or to put it in simpler forms, we have to make a product which is not a copycat, which is similar to my innovator product, which may be slightly different in few of the aspects, ultimately not leading to any clinical difference. Now, the onus of justifying the nullifying the effect of the changes lies with the manufacturer. Say it's a full set of tests which are required. It is in the industry, and from the regulatory perspective, the word CMC, chemistry, manufacturing, and controls, is used to define this. Now, in terms of biosimilars, there further for the biotherapeutics, there are two further categories: the microbial based and the mammalian based. So microbial based are the ones where E. coli or Pichia pastores or Hansenula, these are the hosts which are used for the fermentation. So here the main thing is the post-translational modification is absent. So there is no post-translational modification. But in case of the mammalian based products, because these drugs are specific to each receptor, and the PKPD, pharmacokinetics and pharmacodynamics. Pharmacokinetic is when you take any drug, how fast it moves through your body is pharmacokinetics, and what the body does to the drug is pharmacodynamics. So it means if I have to make a drug which is similar to my innovator, we need to have the same post-translational modification. Otherwise, the response in the patient will be different and clinically we will fail. In order to make you understand this thing in a simpler way, a microbial product is just a pizza base, while a mammalian product is a thin crust pizza with cheese inside and the multiple toppings along with noodles. So, I mean, in terms of complication, I'm just giving you that perspective for easy understanding. Now, just to brief you how this process starts. Once we have our clone is there, we have a thing called cell bank. So that is the heart of any industry. So all the manufacturing happens from a cell bank. So the cell banks are taken out from a -20, -80 freezer, or from liquid nitrogen in case of the mammalian cells, and then it goes into a flask. So this is what we normally do. Say we maintain cultures in the lab, say people, microbiology people, the biochemistry, we maintain the stabs. So the glycerol stocks, what we maintain as the banks. So the bank is taken, it is revived, it goes to a flask, and from flask it goes to a glass bioreactor or a fermenter or a tissue culture flask in case of a mammalian thing. And gradually the seed is increased up to a level whereby it goes to a production bioreactor. Now in the production bioreactor, based upon a particular inducer or based upon the different parameters which we have built into our process, the production of our protein of interest starts. So that is called the upstream. Now what we are trying to do here is we are trying to amplify the protein of interest. Once this thing reaches its, maximum, that fermentation time is also fixed based upon the process knowledge. Otherwise, if there is cell death, that will lead to the introduction of toxic materials, the host cell proteins. So accordingly, the harvest time is also fixed in a process. Now for the mammalian cells, mostly the CHO cells, the Chinese hamster ovary is used, and here the product of protein of interest is secreted into the, you can say, broth. It is externally secreted, while in case of E. coli, the mammalian microbial based products, it is stored within the cells in the form of hydrophobic bodies called the inclusion bodies. So depending upon if the product is outside or inside, the cells are broken open or the supernatant is collected, and then it was the downstream processing. Now the downstream processing is you have your crude product. Now you're trying to polish it, you're trying to refine it. So it undergoes a series of steps, which starts with, say, affinity chromatography, then maybe anion exchange, cation exchange, hydrophilic interaction. Then it undergoes, say, the viral neutralization step is also there, because for most of the mammalian products, the media components which are used, the feeds which are used, so we have to be sure that these are free of viruses. Now, once the product reaches the terminal stage, then the diafiltration is done. So my protein of interest needs to be in a buffer whereby the product is stable for the desired shelf life. So accordingly, the buffer exchange is done. And once the buffer is exchanged, so it gives us the final product, which is the drug substance, or in case of the pharma terminology, it is called as the API, active pharmaceutical ingredient. Once we have that, then it undergoes further formulation, which is diluting it to a particular concentration or adding a particular excipient to give it more stability, followed by the containerization, which is the filling part, either filling it into a vial, into a cartridge, or into a PFS. And say, for the case of biologics, say the process is product and product is process. If you see normal pharmaceutical product, so there the formula is given for maybe 1,000 tablets, and if you want to increase your batch size to 1 lakh tablets, you just need to multiply everything by 100 and you get the output. But in case of biotech products, it is so specific that if you want to scale up your process, then it is very difficult, sometimes even going up in a twofold, say going from 100 liter to 500 liter or 100 liter to 1000 liter. That becomes impossible, and you cannot replicate the process and the product. And another thing is, we need to have a strong understanding of the process as well as analytics. And the three favorite words in industry are risk. Risk is the main thing. Then is the CQA, critical quality attribute, CPP, critical process parameters. So, in order for you people to understand in an easier way, critical quality attribute, each one of us has a critical attribute. Say, the moment you are pursuing your graduation, your master's, some people say, I want to be a scientist. I want to have the maximum number of publications, international publications. Now, in that case, your critical quality attribute is you want to be a scientist who is well known in the scientific community. Then you will not go behind going in for MBA, or if you want to say, okay, I want to settle in early, whereby I complete my master's and I want to settle in terms of X amount by so and so time. Then your critical quality attribute is time, money, or maybe fame. So similarly, in case of a process, any parameter of the product which impacts the shelf life of my product is the critical quality attribute, and any parameter of the process which impacts the critical quality attributes are called as the critical process parameters. And any changes in the process, it has huge impact on the net outcome of the product. Now, if we see in terms of innovator, how the cost factor comes into play, the major cost incurred on any product, innovator product, is the clinical trials. So if you see this slide, you can see two triangles or two pyramids. In case of the biosimilar part, the pyramid which is on the left side, the clinical trial is at the bottom and it is the minuscule, the least part. But if you see in case of the right, in the case of the pyramid for the innovator product, clinical is the most important part because there you don't have any reference. So you need to, based upon the literature, based upon knowledge, we need to develop a product targeted towards a specific receptor, and we need to prove it in human trials. If it passes, it's fine. Otherwise, if it fails, then your entire effort, money, project, everything is gone. So in case of biosimilars, the main onus is analytics, because as I told, the primary, secondary, tertiary structure. So analytics is the most critical thing for the success of biosimilars, since everything needs to be evaluated from those perspectives. So as we go ahead into the presentation, so we need to keep a tab on the primary structure, secondary, tertiary, and what all parameters can help us assess the functionality of the that particular molecule, the potency assays. Now, this particular slide is a very, good glimpse of how analytics impact the overall biosimilar development. Say it starts right from the clone. The clone is the heart of any process. And as I told, say post-translational modification is the most important thing. So if my clone is not properly developed, my post-translational modification thing will go haywire and will never be able to meet the innovator profile, and my PKPD will fail. So another part, when it comes to QC, QC does testing of all in-process samples, the drug substance or the API, which I mentioned earlier, the drug product, all the stability indicating, the stability testing, stability indicating parameters. So the testing at the clone stage, testing at the QC stage, and in terms of regulatory filing, we need to submit data as per different requirements. So analytics impacts there also a lot. Then next, coming to stability. If I don't have the correct analytics for assessing the stability of my product, it means I am going to either make a product which will deteriorate very fast, or if my methods are not suitable, then I may land up in developing a product which may not be feasible in terms of a shelf life. Say it goes off very early. The moment a batch is released into market, it fails. And another major thing is in the upstream and downstream. So upstream feed strategy. So once the upstream starts, once the cells, so each one of us studied the log phase, lag phase. So similarly here also, the phase is defined when we need to start the inducer. So if you see holistically, right from the clone to upstream, downstream, QC, stability, formulation development, and regulatory filing. So analytics is the only thing which makes or breaks a biosimilar. Because say if your method does not say that your in-process sample is failing for a particular impurity, manufacturing people or the R&D people who develop the processes will never tweak the methods. They will say, okay, this is good enough. So it's for us to have the correct battery of methods, as well as to be sure that we are going in the correct direction. Now, as I told in one of the previous slides where the pyramid was being shown, so there's a whole battery of sets which are called the characterization tests, which include the primary structure, general properties, impurities. So this complete battery of sets, the product is tested by R&D through and through. And from these tests, there are few parameters which once fixed will not change drastically. Say if your clone has been fixed, until and unless there are stability issues with the clone, nothing is going to happen to the clone, because you're going to store your cell bank in controlled conditions with real-time online monitoring. So it means based upon the process knowledge, if we do, say, maybe as a vague number, if we do 100 tests in the characterization, out of those 100 tests, depending upon the product and the process knowledge, we say that these 10 or 15 tests are the one which will be used every time for releasing a batch. These are called the release tests. So these are the tests which QC tests every time on a batch for releasing a batch into the market, in addition to the tests which are recommended as a subset of the characterization, there are few tests which are required by pharmacopoeia to be performed, which are the safety tests, like the test for sterility or a bacterial endotoxin or subvisible particles. So there are few other tests which need to be done irrespective of the nature of the molecule or the product. And if you see here, say the precise and accurate is written there, so that is the main, because if we don't have a control on our methods, we'll never know which way our process is drifting and where we are landing. And in case of this release test, if you see the last, the validated one, validation is another thing which is the heart of this biopharma industry. Validation, say if I say as a layman, when you leave in the morning, say if your place, your office, your university, your college is 10 kilometers away, right? And you daily go by a car or maybe a two-wheeler. So you have validated that if I start at 8:15, I will reach there at 8:45 and my lecture is at 9 o'clock. Say if your tire punctures, that is going to happen in lesser number of cases. So you will say, if my tire punctures, I still have a 15 minutes buffer. That is risk assessment. Say a thing what can go wrong. And as a part of this, you get the maintenance done for your vehicle. You say, I'll go in for a tubeless tire. It means you anticipated a risk and you devise the procedure or method around it. So if it fails, then you should have the minimal impact. You means the outcome. Say if you are moving towards if you are going to a lecture, if you are going to your college, so there the intended purpose is reaching on time. So in the process and analytical perspective also, we evaluate and consider everything from the same thing. Now, this is a very good representation of the whole process when it comes to testing. So what are termed as the release test, what QC does every time during a batch, is just the tip of the iceberg. Say if we say there are only 15 tests which are done for a product, or maybe if you see any innovator COA, there may be 15 or maximum 20 tests which are done as a part of lot release. So that is just the tip of iceberg. So actually, if you see the number of tests which go for developing a product and even maintaining a product life cycle, what industry needs to do is, as a manufacturer, what we need to do is we need to trend our batches on a regular basis to even check the drift. Say my incoming materials are regularly changing. Now my equipment are wearing out. So we need to have a plan in place whereby equipments stay within the permissible ranges, they perform as required. So we keep on regularly trending, regularly doing the analysis or interpreting the data right from the process to the reference standards and to whatever parameters we are doing on routine basis. Even analysts, if a particular analysis is failing, we need to go down to the root cause. Is the problem with the analyst? Is this analyst who is, say, wherever he does the analysis, does it fail every time? Or is the problem specific to a particular method? Say if any analyst does that analysis, it fails. And another thing, if you refer underneath orthogonal methods, orthogonal method means any method which you can say verifies or assures a result. Say for easy understanding, if I tell you, if we run a SDS page, so there the separation occurs on the basis of size. Sizes, I mean, based on the size, the SDS molecules attached to the protein, and accordingly the negatively charged protein, it moves towards the positive electrode. So, but if we are having two proteins, right, of the same molecular weight, say 20 kd, so we'll not be able to differentiate it on a SDS page. So then comes the requirement of differentiation or assigning specificity, which is done by a Western blotting test. So in this case, for easy understanding, orthogonal test is a Western blotting test, which is confirming my specificity, that my protein A is this and my other protein B, which is similar in size, is still not the same protein. Now, this slide gives a complete details of tests, battery of tests which need to be covered. And these are few tests mentioned for the, sake of information only. Say the general properties like pH, osmolality, subvisible particle, or the identity tests. So the pharmacopoeial requirement is each product at minimum has to be tested using 2 tests for identity. Say we cannot have a single test, or it can never happen that we have no test for identity in our release certificates. Say if any one of you has got an opportunity to go through any pharmacopoeia, maybe Indian pharmacopoeia, European pharmacopoeia, and if you go to the monograph of any of the salts, say sodium chloride, sodium hydroxide, there are few tests which are mentioned for the identification. It means I need to be first sure that the whatever material I am using, either as a raw material or whatsoever is going out from my premises as a product to be delivered to the patient, is it the same or not? Say you take the example at simple test, simple thing. Say if you happen to go to a fast food and you order noodles. So a person who is a strict non-onion eater, right? So there is another person who is an onion eater, but a veg, and the third person who is a non-veg. You imagine the situation if this identification label goes off. Say the person who is a strict non-onion eater, he does not even eat onion. If he eats that, so the gravity of the thing goes very high, but in this case there is no clinical implication. But you imagine the situation whereby a person has to be given a drug, some other drug, and we give a wrong drug. So it may lead to adverse effects or may even lead to death. So anything which is coming into the as a raw material and is going into the product has to be tested for all the things, and identity is one of the essential elements. Now another thing, if you see any product, it needs to be analyzed for purity. There are different types of impurities which exist in the different the biopharma molecules, and SDS page I think is the most common thing which all of us do during the academics. So HPLC, seldom if a few people are from chemistry, analytical chemistry, and that too they don't get a chance to do it hands-on. So just to correlate in terms of the aggregate impurities, why these are important. So we always talk of there are something called as soluble antigen. So in the lab we used to do one experiment. Even I have forgotten that word. So we'll take soluble antigen in one syringe and we'll add adjuvant. We'll keep on mixing those 2 syringes so that the adjuvant is mixed, and then we'll inject into an animal. Now, whenever anything is soluble, it is not taken up by our body, our reticuloendothelial system, as a foreign or as an antigen or immunogen. So it is not processed and it is cleared from the body. So it means some size, there has to be some size of the molecule to give impact. But at the same time, if my molecule is showing aggregation because of several parameters, because of stability issues, because of some structural deformity, if those aggregates increase, then my body is going to take it as a foreign thing and antibody will be formed against that aggregate. The moment next time any person is injected the same drug, there will be a clinical effect or a shock sort of thing which will appear in the patient. So there are other battery of tests which are mentioned, like high order structure, surface charge, which needs to be done additionally. Now here I have shared one profile. When I say that it's a biosimilar, say for a manufacturer to claim a product as biosimilar, that product should be registered in that country. So that is why I can claim. Say if nothing is available in India, so I need to define what I am taking as a reference standard. If I am taking a EU, European Union reference standard, or I am taking the US as a reference standard. So normally, the majority of the area, the regulated markets, what you say in industry is the ICH. The ICH region comprises of US, EU, and Japan. So those are the, say, three stringent areas or the regulated areas where the regulatory requirements are very well defined and very stringent. So the depending upon the intended market, say if we say US is the biggest market for this particular drug, I am going to make a biosimilar against this. Then accordingly, we need to select the innovator product, and then we need to do our say complete process has to be designed and everything. Now this slide just gives a comparative thing. So as I told, we need to prove structurally it is similar, functionally it is similar. So this overlay, chromatogram overlay, it shows the peptide mapping. Say in order to give you a better idea, if my protein is folded, say it has got a secondary structure, tertiary structure, depending upon how it is folded, there'll be certain sites which are free, which are not enclosed within the folded structures. So we take enzymes which will chop off my protein at a particular site, and if my protein configuration is similar to the innovator configuration, so same enzyme will cut both the samples, the reference sample as well as the my in-house sample at the same site. And when we run this using a RPHPLC, which separates it on the basis of hydrophobicity, it will give us this profile. This is another one in terms of the charge variants, that is the acidic variants and the basic variants. So, I mean, this is just for illustration. A whole battery of tests need to be provided and the data needs to be shared. Now, coming to from the biosimilar perspective, how the analytics are most important. As I always say, the only thing which can make a biosimilar successful is analytics, analytics, and analytics. So right from development, the identification of QTPP, this is called as the quality target product profile. That is, if I am taking a particular reference, the innovator product, then what are the key parameters as per which I need to design my process? What will be my critical quality attributes? And the QBD, quality by design, is an approach which you can go through the net. So I mean, each of these words and say subjects has a very deep, I mean, you can go as deep as possible, but just for the sake of understanding, we can say the predefined specifications and considering all the statistical evaluation. Then is the analytics are involved in the biosimilarity establishment. As I told, that we need to prove that our product is similar structurally as well as functionally to the reference medicinal product. Clearance and hold time studies. Clearance means, if, as I told, that as we go to the upstream, my host cell proteins, say I have inserted my gene of interest in CHO cell. So CHO, while undergoing the fermentation, will produce its own metabolic machinery, and it will produce its own molecules, so own proteins and other molecules, which includes host cell proteins, DNA, and there are other contaminants also. So clearance study means, so once we harvest the batch, suppose if I have got the host cell proteins at 10,000 nanograms per ml at the end of upstream, and the desired value in my drug product or drug substance is 10 nanograms per ml, then we need to design a process that how much capability my process has to remove these impurities. Say, can it remove from 10,000 to 10 or 10,000 to 1? And clearance means as we are gradually going to the downstream final stages, my impurity should get removed without a loss of my protein. Now, loss, I'm not saying 100% efficiency can never be achieved, but around 60 to 70% efficiency is what exists for majority of the downstream processes. And now with the advancement in techniques and with the advancement in column chemistries, there are several options available in the market whereby we can work on the cost. Another factor is analytics come into play as a reference standard. Reference standard, because say, as I told, that we need to prove that my in-house batch is similar structurally and functionally to the innovator molecule. So we need to have a calibrator, a particular batch which has been fully characterized, against which we will be doing our release testing for the content as well as potency. Now moving to next, scale up. Scale up means, say today, if any regulator goes to any manufacturing organization, they ask what is your scale. Say if I say that I am a manufacturer of say GCSF, and my current scale is 100 liter, and it's a EU regulator, the European Union. They say we have a huge demand of GCSF. GCSF is a granulocyte colony stimulating factor, which is used for the post treatment of cancer patients. So once anyone is given a chemotherapy, the white blood cells are destroyed because of the radiation. Now people have neutropenia. The neutrophils, they get depleted, and because of lower counts, the person is likely to contract infections and can lead to death. Now this is one of the product which is administered to increase the production of those granulocytes. And accordingly, the regulator says, okay, I see that your process is good, your systems are good, but your scale is say 100 liter, and depending upon my requirement, even if you work all through the year, you'll not be able to meet my demand. So you need to take it to 1,000 liter. So we need to develop a process which is scalable, and whenever we scale up, say when we scale up any process, there may be practical constraints. We may be forced to change the design of a bioreactor, a fermenter, any downstream column, because of the constraints at that particular scale. Now, these changes may lead to certain variations in terms of the process, but the critical quality attributes and my final product needs to be the same. It needs to match at the initial level. Now my reference, suppose I made a biosimilar first, for which I got a marketing authorization against RMP, which was 100 liters. Later I go to 1,000 liters. Then again, the market demand increases so much that I go to 5,000 liters. So I need to show a comparability that my 100 liters, 500 liters, 1,000 liters, and 5,000 liters, everything is the same. So this is what is the comparability study. And in terms of the manufacturing process, it has to be continuously monitored, and within the process there have to be critical stages, which is a go no go stage. Say there are few process adjustment steps whereby if a parameter is drifting, we can adjust. While contrary to that, there are other stages. If we don't meet a specification, my terminal product is going to fail. So in that case, we check regularly as per the defined interval and stages, and accordingly the lot release takes place. Now, it's not only, from the process perspective, but from the analytic perspective, how it is all done. So first is the literature survey. Second is the risk assessment. So nowadays, organizations mostly work on platform technologies. For easy understanding, say, today if you are, say, planning to open a restaurant, you prepare gravy, you put peas in it, it will become paneer gravy. You put grams in it, it will become chana gravy. You add, so your gravy remains the same. So this is what platform technology means. If you're working on protein A chemistry, protein A column will remain the same. Only thing what you need to tweak is the charging density of your protein depending upon your molecule or the process specific data. Then for establishing the limits, say, since I know the terminal thing, how can I match the innovator? Say the innovator COI is not available to us readily. So we need to test multiple lots of the innovator. Now another variable is innovator does not provide us the methods what they are using. So we need to test using the methods which are available for us. Based on that, we need to define the specifications for our product. Then we need to develop the methods for the intended use, and we need to validate them. Intended use means, say, if there's a product, we all have seen vaccines, right? The oral polio vaccine, everyone might have gone to get the dose administered to their kid, nephew, niece. So if you see there's a vial and there is a VVM, vaccine vial monitor, whereby you check the color. It means if the product has crossed that particular time, it has degraded and it will not give you protection, or it will not generate antibody once the vaccine is administered. Now, if we are testing this product, but my method is not capable of failing it. So it means that that particular method is not fit for intended use. So we need to spike, we need to test the product which is passing, then we need to degrade the product, maybe using pH, maybe using temperature or any extreme conditions to check the process, my analytical capability, that is my method capable of detecting the product failure or not. Then everything needs to be validated as per ICH. So ICH is the gist of pharma and the biopharma industry. Now, another thing is only understanding the protein stability and the protein denaturation pathway is not important. Another thing what we need to consider is the process feasibility. Say, if my molecule is so labile, it is so sensitive that we cannot even produce it in the manufacturing conditions, so it is of no use. So during development, we need to understand the interdependency. Say, what is the constraint? What is the, say, if my protein of interest has a particular amino acid which is prone to oxidation, if the headspace in my vessel increases, the degradation is going to increase. Then I need to have a strategy, because if I make something which cannot be scaled up or which cannot be manufactured, developing such a product is of no use. Now, finally, I mean, in terms of the analytics, how the analytics are important, since the biotech products are the very complex molecules, so the batch to batch consistency needs to be maintained. We need to keep a check on the reference standards and the analytical method performance. If my method starts behaving abnormally and I'm not trending, right? Just to give you a simple example, if I am having a weighing balance at home, and if I have a tendency to gain weight, I take my weight today and it's say 50 kg. I keep on eating too much and I am not doing any physical activity. Still my balance is showing 50 kg, although there is a change in my physical appearance. It means my method of measurement is drifting. So same is the way what we see in the QC, that how my method is drifting, and if whatever failures we are getting, what is the cause of that failure? Is it due to the process? Is it due to the method? Is it due to the analyst? And that is what is called as kappa, the corrective actions and preventive actions. So these few things like kappa, change management, kappa, effectiveness, these are the heart of any pharma and biopharma. And the product and processes are monitored and trended. So this is what the regulators also see. What is the control on our process? How do we track that? And kappa effectiveness monitoring is the pulse of a quality system. Just to make you understand the concept of kappa in a layman's language, kappa is a corrective action, preventive action, right? Say, if you have a house on the third floor of a building and you have a kid, a small one, maybe 3-4 years, who has 3 years, and your balcony railing is very low. The kid comes running, tumbles over the grill and falls down. He's hurt. The kid is bleeding. You take the kid to hospital and get the first aid medication done. That is the correction. It means the problem what you encountered, you tried to address that problem. That is correction. So when you come home, you find root cause. Root cause was you cannot stop your kid from playing and running. So you increase the height of your grill. Root cause is the lower height of the grill is the root cause. So you increase the height of your grill. That is your corrective action. Since your entire colony, the same design of grill was put by the builder. So there is a potential today your kid has fallen, tomorrow some elderly or another person can fall. So as a preventive action, you reach the secretary of your society, you approach him and you try to get all the railings, additional railings installed to ensure that someone does not accidentally fall. That is preventive action. So that is what we routinely follow in industry so that we don't get failures repeatedly. Now, in terms of the analytics, what we have all studied, it's not only the manufacturing of the drug substance, but also the drug product. Say the selection of the container, what is the compatibility of my container with the product? Is something leaching into my container? Then you might have seen if you bring some drug at home, it is written do not freeze. What if it freezes? Say another thing, if something has freeze, I mean, how much multiple freeze thaws can it undergo? and say if 228 or maybe a -20 drug was accidentally exposed to a room temperature, maybe 25, or I mean, it melted and it again froze. So we need to be ready with all these studies to address any failures and as a part of the regulatory expectations also. And another thing is the preservative efficacy. So if you see anything as a multi-dose container, then preservatives are added to those drugs to avoid the growth of entry and the growth of organisms in the container. Say due to each prick, the moment we prick to withdraw one dose, and if there are multiple doses, so we need to be sure that there is no contamination which exists in that particular container if it enters accidentally. Now I would like to conclude my presentation. And it's a say from the biosimilar development perspective, it requires to design the process as well as analytics. We need to match the innovator parameters, and most important thing is we have to be cost effective. If we make a process which is say we today I say I'm going to design something which is cheaper than this Tata Nano, and I, because of my limited resources, I make a car which is say for 10 lakhs, for which multiple options are available in the market. So my product is never going to sell in the market. And at the same time, we need to consider the considerability, the scalability, sorry. And as I have been discussing through the presentation, the heart of biosimilars is analytics. It's analytics only, the testing only, which keeps you on track and into business. If analytics fails, everything fails. And the main gist is the process is the product and the product is the process. So anyone who is a party to any of the smallest activity needs to be aware that if something is going wrong, it needs to be addressed, and the accountability part is the most important thing. And I would like to conclude my presentation saying that an approved biosimilar can impact the life of billions or billions of people due to the availability of affordable medicines. So it's a great cause to be a part of this particular industry. Thanks for your attention. Questions, please.
Thank you so much, sir, for the very
everyone.
informative and very detailed talk about touching each and every aspect of biosimilars and the process which is involved. Yes, students, now the session is open for some question answers.
Hope this thing did not happen during the session.
Yes, sir.
Yeah, any questions? Can I stop
How
sharing
long
the
is-
screen
Yes,
now?
sir.
So you can stop. Yes, sir, you can stop the presentation and then they can ask.
Sure, sure, sure.
Excuse me, sir. Sir, how long it takes to develop a biosimilar, and do biosimilar need clinical trials also?
Say if you see innovator takes around 12 years to develop, and biosimilar, as I told, the clinical trial is it's a small one. It's a very smaller version as compared to the innovator, and depending upon the drug or the molecule, sometimes the waiver means the waive off is also there. And say another difference is in case of an innovator thing, the phase 2, which is the dosing study, that is not applicable in case of a biosimilar. But for a biosimilar, minimum 5 to 6 years is what it takes to bring it to the market, because you need to develop everything. So accordingly, 5 to 6 years is the minimum period what it takes. So everything needs to be calculated back. If a particular innovator is going off patent in say 2030, then we need to start in 2024 for that. Hope I have answered your question.
Yes, sir. Thank you.
Yeah, welcome.
Yes, students.
Sir,
Excuse
can
me,
a
sir.
biosimilar of a drug be whose patent hasn't been expired yet be used for emergency purposes?
Pardon, can you come back again?
Sir, can a biosimilar of a drug whose patent has not been expired yet be used for emergency purposes?
No. Say the innovator company will sue you, and they'll charge millions till the time that patent is there. Until that patent goes off, you cannot sell. Even if you sell earlier by 2 or 3 days, they file a lawsuit against you.
Okay, sir.
Sir, do biosimilar works in the same way as biological reference products?
Exactly same. So that is the very purpose. That is why the thing of switchability comes. Say, to give you a simple example, today if you are using a brew filter coffee, right, and your taste buds are used to a particular taste, tomorrow some another thing comes, say lata coffee comes in the market. The person says this is a very good coffee at 1/3 the price, but are you going to compromise on your taste? Even if you try once, you'll say no, 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 I am not used to this sort of taste. I'll spend more. Similarly, to convince a particular doctor or a patient on the switchability can only be done if we have that data. If there are adverse effects arising from a biosimilar, then no one will take it. So all those things are taken care by the regulatory agencies. Based on that only they give the permission or the marketing authorization.
Okay, thank you, sir.
Yeah, welcome.
Sir, as we know that production of a vaccine or any medicine takes so many years, and we are getting news of coronavirus vaccine, how did it manage to come so quickly within a year?
So based upon my experience, what you can, if you remember, there was a swine flu, which I think came in 2012, if I'm not wrong, 12 or 13. And then there was another SARS, which came in 2000, again 14 or 15. So all these diseases were mRNA based virus things only. So this swine flu virus vaccine also came. That time the vaccine was prepared considering this, because that also came, I mean, extended over a longer period. Although it was not a pandemic, it was epidemic. So the manufacturers had already had that technology. Now when this thing came, the base was already done. The platform technology, what I told Gravy, was already there. And based upon so many, say, since it was a push from the organizations like WHO, so all the people who were initially competitors, they came together. So because of that, I mean, the things happened so fast.
Okay, sir.
Yeah. Any other
So what
question?
is between biosimilar and generics?
Pardon?
What is difference between biosimilar and generic?
Okay, I'll explain you in a simple layman's language. Say, if there is an essay, a printed essay, you photocopy it and you take a photocopy of that particular page. That is a generic drug, right? And if you take that page, you read it and you write in your own language without changing the meaning of that particular essay, that becomes a biosimilar. So it means it will not be identical to the original thing, but the clinical effect will be the same.
Okay, sir.
Sir, does this mean biosimilars do not have to undergo clinical trials because the clinical effect will be same?
No, biosimilars undergo clinical trials. It has, say, different, say, phase 1. Phase 2 is not required for biosimilars, because phase 2 is a dosing study whereby it is standardized how much dose is to be administered to a patient. The innovator has already done that. In the phase 1 study, the PK/PD, immunogenicity, everything is studies. Phase 3, say, depending upon the molecule, say, I'll give you a simple example. If it is a molecule which is meant for, as I told, GCSF, for neutropenia, if you take any human being from any geographical location, say maybe it's a Indo-American, Red Indian, Asian, Japanese, the moment you give radiation to any human being, radiation is always given on the weight basis, on the basis of body weight. So the killing of white blood cells will happen the same. So if you have done a small study, you don't need to go to a global study if you want to go to a particular geography or to any European country, right? But if you see the cancer, the liver cancer, which is caused by the drinking of alcohol and smoking, the way the smoking and drinking habits what exist in India will not be the same what exists in Latin America, because there the drinking pattern is different, the tobacco what they use is different. So depending upon that, what is the, basic course of development? So that varies. So in that case, it may be required, it may not be required. So the regulatory agencies evaluate that. So the manufacturer puts our perspective, say this is based upon my evaluation and my risk assessment. Then there are scientific advices, because the regulatory agencies also support the release of biosimilars. So based upon the information and data shared by manufacturers, they have the subject matter experts. They charge for it and they suggest you for that this is the approach what you can use. Accordingly, they give a permission that you can go in for a waiver, or you need to do a study. If you need to do a study, what should be the minimum number of subjects, and if it has to be, say, only a India only study, or if you want to introduce across the globe, has it to be a global study? So depending upon the molecule and depending upon the disease prevalence, how it progresses. So that is the basis of designing that.
Yes, girls, any more questions? Deepika, do we have any questions from the YouTube? Dr. Deepika, do we have any questions?
Yes, ma'am. Yes, sir, we have question on YouTube as well.
Okay.
So the question is, what were the trials done for proving the efficiency of the COVID vaccine?
Say, I'll not be the correct person to answer it. So since I am not from that industry, and as such, I am not representing myself as an industry person, I'm just sharing my opinion. So whatever is thing in terms of a viral neutralization, so that is the thing. Say, basically vaccines, say, all microbiologists study, as per my understanding, the vaccine part as a part of curricula. And since I was in vaccine industry, there are three different approaches: active challenge, seroconversion, and neutralization. So since this is a virus, so neutralization approach, say the generation of antibodies, and there's another concept called, if I remember correctly, because it's been almost 20 years now I left the academics, premonition. Say, once a virus occupies your receptor, then other viruses cannot attack that, which are related to it. So there may be several factors which I may not be able to answer correctly or means accurately.
Okay, sir. And sir, there's another question from the same person, Sahaj Kakkar, that why do we need a biosimilar one? If one is already that drug, why that another kind of drug is required?
Say, if a person has a disease, say if I need a drug, if my name is Sandeep Tata or Sandeep Ambani, I can afford an injection which is 10 lakh. But if I am Sandeep Pathak, I will look for something which is 50,000. And if I need 10 injections, I don't have the privilege and to spend 1 crore if I can get the same treatment in 5 lakh. So I will just look into it.
Okay, sir.
So it's the economic paying capacity and the ease of reach, because now the cancer and the cardiac things, 2 out of 3 people are now having cancer issues. So more and more biosimilars are targeted towards different type of cancers. So simple thing, say, as I gave the example of GCSF, there is another product, PEG GCSF, which is the same molecule covalently linked to a chemical. So in today's date, if someone from US takes 1 PEG GCSF injection, so the amount spent there is more. If the person comes to India, take a biosimilar, just tours for 1 month and goes back, that expenditure is lesser. I hope that thing will make it easier for you to understand.
Yes, sir, sure. Thank you, sir. These are the
Yeah.
questions that we have got on YouTube. Yes,
Yeah.
Sandeep, ma'am.
Oh, yes, I think Chelsea, you want to ask something?
Yes, ma'am.
Yes.
Sir, you are yourself a part of industry. So how should we go about a career in the industry in this particular field?
The thing is, first thing is, whenever you get a chance for the industrial training, nowadays you people are very lucky. There are smartphones. You can get everything at the click of a button. So you put a root word, it is there. And say, my personal advice is, we spend so much time on Facebook, this WhatsApp. So if we spend at least 4 hours for ourselves, just for getting this, say, all these social media links and looking for ourselves, there are multiple options. Say, you can go as a part of, there is a BCIL thing where you can approach for industrial trainings. And nowadays there are several private organizations who provide you, say, training for maybe 1 month or something. They charge you for that, but you can do hands-on analytical equipment training. So there are several options. And the only thing is, say, if you want to really join industry, the first shackle which you need to break from your mind is that I'll stick to this particular city. Say, I am from Chandigarh, I have moved to 5 different states. So if you say I have to stay here only, that is the biggest barrier which is going to restrict you. Then if you are willing to move places, I mean, there are ample opportunities everywhere.
Sir, we'll appreciate if you could share few links for the industrial training, because we have industrial training as part of the curriculum, and I don't think these girls are of that mindset that they will stick. They're from all backgrounds, so I think they're willing. Yes, girls.
Sure. I can help. I'll share the details. So the only thing is, say, there are industries close by also. Say, but I tell you one thing that, say, I am a microbiologist. Sometimes people ask me how come I am into analytics. Because, say, if you go to a hardcore pharma, a microbiology is a person which is just a subsection of a lab. Say, microbiology is one of the sections. But if you go to the injectable side, the therapeutics and the vaccine side, so this is the core demand. So only thing is, one is the awareness. Then, I mean, if you get more understanding, say, within the industry, there are several, what you say, divisions, say, quality assurance, regulatory affairs. There are so many courses also which you can pursue after your, say, completion of masters, or along with your even job also. But I mean, I can for sure share some details. The industries who offer this industrial trainings. But the only thing is, you need to move out of the place and you need to go to places. That is the only thing.
Yes, for sure. Yes, girls. Any other question?
Yes, Sandeep, we can have a pic with sir. So
Yes,
if
sure.
Sure.
Girls, please put on your camera. Switch on your cameras. Deepika. Good.
So all our studios, majority have spectacles. Is it done?
thank you, girls. Yeah, thank you so much, sir.
Thanks a lot. Any other further questions from your side?
Yes, girls, any questions you wish to ask, feel free. कोई silly question भी चलेगा.
Sir, kindly tell us about biosimilars.
Say, biosimilars, as I told, say, there are several drugs which are available in the market. Say, I'll tell you one simple example. Say, previously it was told someone has diabetes and the person became blind. In old times, they used to say, वो बंदा मंदिर गया, उसने संतोषी माता को दही से नहला दिया, रात को अंधा हो गया. That was never the reason. The thing was, the person was diabetic. Because of diabetes, the retina was getting detached. So nowadays there's a drug, and if you go to the innovator drug, the cost of injection, one injection is 1,75,000, and say it almost requires 5 to 7. But if you go in for an innovator brand, so it's maybe within the cost of one injection itself, you can complete the, I mean, full treatment also. So, I mean, just to put it in simple words, it's the same clinical effect in a cheaper version. So cheaper will not be the correct economical version. Cheaper, when we say, it comes to the other way around, that the quality is substandard, but quality is never substandard. So everything is evaluated, and only then it is released into the market.
Sir, after the biosimilars are in the market, so as both of them have the same functions, but the biosimilar is available in lesser cost, doesn't the patented person, the innovator, go bankrupt after the release of biosimilars?
No, they also decrease their cost, and because their R&D is so strong, they keep on introducing new products every year. Say, I mean, innovator R&D is so strong So they have almost so many products in the pipeline, they keep on introducing one or the other product. And even in terms of patents, there are several patents. I mean, it's a product patent, then a process patent, then a formulation patent. There are several patents which can be extended. Say you take the simple example, say, I mean, if anyone buys, say nowadays Mercedes is available, then you take I mean, limousine, Lamborghini, you put anything. Although the lesser numbers are selling, so worldwide they look at their target and say, based upon, if business is not feasible to a particular innovator, then they withdraw from that particular market. That is a more of a business strategy.
Okay, sir.
Yeah.
I think any other questions, or is it over?
So if there are no further questions, then
Yes.
I think it's good to go off, and I would thank everyone for patiently listening to me. And I hope I was able to explain the subject and to answer your queries whatsoever you asked. In case if still you have some queries, you can share your queries with the coordinator committee or with Sandeep. And if, I mean, whatsoever things I can answer, I'll try to answer those.
Thank you very much, sir. And really, we appreciate, first of all, for your consent, and secondly, for your time. We understand your schedule; it's really tight, and we will appreciate for more future events and talks from you.
Sure. Thanks a lot. Sure.
Thank you so much, sir.
Yeah.
And on behalf of MCM DAV College and our department and all the students, I extend wholehearted thankfulness to you, sir. Thank you. Thanks a lot.
Okay, thanks
Thank
a lot,
you, sir.
everyone. Thanks a lot.
Thank
Bye. Thanks.
Thank
Bye.
Thank you so much, sir.
Yeah, thanks. Bye.
Thank you, sir. I think Deepika, you can take over.
Yes, ma'am, sure. Has sir joined us?
Yes, Deepika, he has joined.
Okay, ma'am. Okay.
Good evening, sir.
Good evening, ma'am. Hello, am I audible?
Yes, sir.
Yeah.
Yes, sir,
Good
you're
evening,
audible. Yes, sir. Good evening, sir. So, good evening, everyone. Thank you to each and every one of you for continuing to be here with us today and who are sincerely committed to this event to make it a success. So, now I would like to welcome our next speaker for the occasion, Dr. Dheeraj Kumar Nanda, who is currently working as Assistant Professor at Asian Institute of Public Health, University, Bhubaneswar. He's an MSc and PhD in biotechnology. He has also done postgraduate diploma in intellectual property rights and in clinical biochemistry and medical biotechnology. His major research functional areas are biotechnology, microbiology, molecular biology, nanoscience, bioinformatics. He has worked as a research associate at ICR National Dairy Research Institute, Karnal. He has also worked as senior research fellow at ICR National Bureau of Animal Genetic Resources in collaboration with NDRI, Karnal, for more than 5 years. He has also worked as GRF at Central Tassar Research and Training Institute, Ranchi, in collaboration with Department of Biotechnology, IIT Kharagpur. He has also worked as a lecturer at Marwari College, Ranchi University, and as a guest faculty at Bisra Institute of Technology. He has also been awarded the Best Poster Award in the 9th National Symposium of Society of Conservation of Domestic Animal Biodiversity at Pune in 2012. He has also acted as a reviewer in many esteemed journals like Process Biochemistry, Journal of Microbiology, Biotechnology and Food Sciences, and many more. He is also patent holder for his study on a cross-linked membrane with flow line capable of arresting free-flowing gold nanoparticles and the process for the same. He has also submitted a total of 159 DNA sequences to NCBI GenBank. He has published for more than 16 research papers in various renowned journals, along with 4 papers and 13 abstracts in conference proceedings. So we welcome you, sir, for gracing the event on behalf of each and everyone. Thank you, and we are looking forward for a lecture. Please, sir.
Yeah. Thank you, Deepika.
Welcome,
So, thank you for my introduction. Now I will put my presentation. So please.
Yes, sir.
Yeah.
Yes, sir. Please go ahead.
Yeah. I have. Is it visible?
No, sir, not yet.
Okay.
It is visible, sir.
Okay. Thank you. So, yeah, just a minute. So my presentation will be on COVID-19, and just I will give an overview because I am also not an expert of this virology, and I am not also expert in the drugs which have been developed for this COVID-19 or vaccines which have been developed. Just as a microbiologist and whatever knowledge I have got or the information which I have collected from the internet and the resources, on the basis of that, I have prepared my presentation, and I will be just giving a output what actually this COVID-19 is and how the drugs have been developed, what are the drugs, a basic. So it is not in depth, but it is just you can get an idea about this. So this photograph, if you see, I have taken it from the state news website. In this photograph, you will see that the entire scenario of this COVID-19 has been explained in this one diagram. So the journey started that the virus came, then the people, they were scared of this because there was no prevention for this. There was no drug for this. So everywhere was scared. And we then were introduced with different types of guidelines. A huge money loss was there, and many people had to be at home. Then the time came when drugs and the prophylaxis, means the vaccine, came, and we are on the way. When, so you can just read this article. So I have given this link, so you can just go to statesnews.com and you can search for this photograph, and in that you will find the entire story. It is a very good story which has been written for this COVID-19. So this scenario is going on, and the drugs have been developed, and still the path is there, and we are on the way. And it took 12 months for us, and beyond this also there is a way where we have to go till we are having a time when the COVID-19 will go from our society, and we will be not be scared of that. The similar scenario came in India also. So it started in March, and in different phases of lockdown and unlock, we were given the guidelines, and if we see specifically for the study, we shifted to a new platform, that was digital platform or the online platform. Even I am thankful for this, that still for due to this platform only we are able to attend this type of we are able to organize this type of seminars like you have organized. And it would not be that much easy to give this talk by going to Chandigarh and then giving this talk. So in this online platform it is easy. So we should be we can be thankful that this COVID-19 case, we learned a lot of things. So at home also we can do this all things. Then the path went on, and various vaccines came, and we are waiting for that vaccines, and vaccines have arrived. And once we get the vaccine, we will be back to our work. And this all is we can say that in nutshell, whatever COVID-19 is doing, we want a cure, and then we want to go to the normal life. So this will be the path where we want to go. Right now, if we want to see what is the condition of this COVID-19 throughout the world, so according to WHO data, just a minute. I think the screen is not there, no? Can you see the WHO data? Hello?
Sir, it's the screen is there.
Yeah, it has. Yeah, it has come.
Yes, we can see it now, sir.
Yeah, voice is clear. Is it audible?
Yes, it is audible.
Okay. So if we see the scenario of this COVID-19 throughout the world, according to the data which has been collected yesterday, throughout the world, means like 883 million people have been affected due to this. And a lot of death cases have been also reported. In India also, more than 10 crores people have been affected by this. And we have loss of more than 150,000 peoples. But if you see the death rate throughout the world, the death rate which is there, it is 2.1%, but in India it is 1.44%. So we are very much lucky, or we can say that we are that much prepared that this type of virus is causing less death in comparison to the other countries like USA. So I'm not going to do any more analysis on this because I am not the right person to discuss this. But as a microbiologist, we want to know what actually this COVID-19 is. So if we see this COVID-19, the name COVID, what actually this denotes. So this COVID, it denotes CO. If you split it, so it comes coronavirus disease 2019 because it emerged in 2019. So WHO has it has given a manual in which the nomenclature of disease have been given. So according to this, the World Health Organization best practice for the naming of new human infectious disease, which was being published in 2015. According to this, the book or the manual or the practice, this COVID-19 was named for the newly emerged disease which occurred in 2019, and this nomenclature was given in the February 2020 by WHO and IOE. Now, what is the virus which is causing this disease? So the virus is SARS coronavirus 2. Why it is coronavirus 2? So the nomenclature of this viruses, it is been given by, you might be knowing that there is a committee which is called as International Committee on Taxonomy of Virus, which is in London. So they give the nomenclature for virus, and they have made this nomenclature in 2020 February. And why they have named this as SARS coronavirus? So the disease which it is causing, it is severe acute respiratory syndrome. So for that, this SARS was given. And this one is not a new SARS. Previously also we had encountered, which I will be explaining later on. So due to that, this was named as SARS-CoV-2 or SARS coronavirus 2. Why this is called coronavirus? So if you see the structure of this virus, it is having some crown type of structure. So in Latin, crown means, in Latin, corona means crown. So this, if you see the structure of this virus, it is having something like crown. So due to that, it is called as corona. And according to the nomenclature or the phylogeny, also it comes under the family that is Coronaviridae. So it has been given the like phylogeny from the Coronaviridae, and it comes under the beta coronavirus. So this is its phylogeny. Then this virus, it is not very much new for us. So it has not only emerged in 2019. Previously also this was there, and in 1900s, this coronavirus, it was known to cause diseases in case of birds and animals, and still it is there, and it basically causes the disease in the upper respiratory system, then external tract, and in some cases it also does the disease in the reproductive system and nervous system. If we see different type of coronavirus which are present, so in humans, we find that there are a number of coronavirus which have previously been encountered, like coronavirus 229E, then NL63, OC43, then SARS. So this is not new. Previously, although we have encountered, and the first incidence of this SARS coronavirus, which was encountered, it was in 1960, which was SCOV229. Then in 2003, this SARS came, which was SARS coronavirus. So that time it emerged. Then in 2012, again similar type of virus came, which only caused this type of syndrome in the Middle East. So it was named MERS, that is Middle East Respiratory Syndrome coronavirus. And in 2019, this SARS coronavirus 2 came. If we see the structure of this virus, how it looks, so the structure is having crown type structures over the surface of it. There are some projections which are called as spike projections, and these are the spike proteins. So if you see this red color spike, they are called as spike proteins, and below that there is a membrane because it is an envelope virus. So it is having an envelope protein, and inside this envelope there is a nucleic acid, and inside this nucleic acid is the genetic material of this virus, which is the RNA. So this is a RNA virus, so it is having RNA as the genetic material. Besides this, it can have this virus, they are having DNA also as genetic material. In RNA also there is plus strand, minus strand. So this is a plus strand, single stranded RNA virus, and the diameter is approximately 100 nanometer. So it can easily pass through a normal bacteriological filter, but if you use multiple layer of filters, then it cannot pass that, and it cannot enter there. So for that, we are using multi-layered mask. So that for that also some publications have been done. And if we see how this virus actually replicates, so there also it is using the system which is present inside our body. So any virus, because this virus, they are obligate intracellular parasites. So they get entry inside the host cell. So depending upon the host who is, they find some type of receptors in the body, in the surface of the host cell. Like if we are humans, so in us there are some receptors, and we have one receptor which is the ACE2 receptor, which is present mostly in our epithelial cells, like in the respiratory system also we are having, in intestine also this ACE2 receptor is present in various cells. So like if you have to go to any place, you or if you have to get entry to a home, anybody's home, you require to have the key of that home, then only you can enter. Similar way, this virus has found this receptor as the lock, and for that key it has made this spike proteins. So in this spike protein there are some receptor binding domains, or RBD, which are some we can say amino acid sequences which are very much specific, where they can just have a similar type of structure like the lock and key, and they can get entry into this receptor and they can recognize it. Once they have recognized, so first is binding of the virus to the surface of the host. So for that they use this receptor. Then they want to get entry. Now for this entry, what they do, they use this endosome formation. We know that in the eukaryotic cell there is endocytosis. So by this endocytosis, this endosome formation takes place and the virus gets entry into this host cell, that is the human cell. Now inside this human, it has to release out its genetic material. So once it has released out its genetic material, so here the genetic material is the plus strand single stranded RNA. Now what is this plus strand single stranded RNA? So it is the messenger RNA which we are having. So this RNA is having the similar feature which we are having, like it is having a poly A tail. So in eukaryotic system, there is a poly A tail in the messenger RNA. So this virus is also having this poly A tail. So it can easily bind to the ribosome and it can do the translation, and it makes its protein. Now the protein which was present over the surface of this virus, they are the structural proteins. Okay, now when it gets entered inside the host and it makes it protein, now this protein are non-structural proteins which are not present in the structure of the virus. Rather, it is only used for to control the machinery inside the host cell to make its copy. Now, to make its copy, it will replicate its genetic material. It will make the different proteins which are required to make this structure. They will assemble themselves and finally they will be released out. Once they have released out, they will have multiple copies of the virus. Now, once the virus has come out, it will be called as virons. Virons means the infective particles which are not inside the host cell. So these virons will come out, and the burst size of this virus is 10 to power 3 virons. So if one virus gets entry into the cell, it can make its thousandth copy and it will come out. So after coming out, it will target the next cell and very fastly. So you can see that the viron entry is in 10 minutes and the eclipse period is 10 hours, means up to 10 hours it is inside the cell, and after 10 hours it will come out in the form of virons, and then it will attack the next cell and it will go on to replicate itself. Now if you see the genetic material of this virus, so it is not a complete mRNA, it is a fragmented RNA. So in this fragment, it is having different ORFs, like there is ORF1A. ORF is open reading frame. So for encoding any gene, there is an open reading frame which is having a start and a stop codon, and in between whatever amino acid sequence has to be made, it can be made by that particular gene. So here, this open reading frame, there is ORF1A, 1B, then there is a spike, then envelope, membrane and for nucleocapsid. So it is having different fragmented RNA, and this RNA can make different proteins like the ORF1A and 1B. They may this 1A, it makes a polypeptide. Polyprotein, we can say. Polypeptide, no, it is a polyprotein in which different other types of protein are also, like there is pepin like protease, which we can further cleave it, and they can cleave and they can make the protein of their utility. Then in this there is in this ORF1B, there is a RNA dependent RNA polymerase gene. So this gene is required to make the RNA dependent RNA polymerase. So because this is a RNA virus, so it has to copy its RNA. So for that it requires to have this RNA dependent RNA polymerase, which it will require. Then for this spike and envelope, they are required to make this spike protein. So in this spike, there is a portion which is called as the receptor binding domain, or RBD, which can recognize this ACE2, which is the receptor which is present in the human cell, and it can recognize and it can bind and then replicate. So for this, this entire machinery is using this all proteins to replicate the virus. So similarly, all the virus, they have this type of mechanism by which they make the host, they make themselves compatible to work in the host cell, and then only they can multiply. Like if you go to anybody's home, if you are feel comfort, then only you can be happy in that home. So in a similar way, this virus, it makes itself happy inside the host by making this all protein, because this was not present in the structure. So he didn't carry it with him, but the virus, when it went into the host, then it made these all proteins, and that it used the machinery of the host cell. So this was about the replication, how it does. Now, if we see the signs and symptoms when this virus gets into the body and it replicates, then we have different types of symptoms, which is very much common. You might be knowing all this, that mostly the symptoms are similar to that of the common cold and the flu. But here, the shortening of breath is one of the symptoms which is seen in this case of COVID-19, which is normally in common cold and flu, it is not present. So by this shortening of breath, one can know. But just by knowing that you have these symptoms, it doesn't confirm that you have this COVID-19. So to confirm this COVID-19, you require to have some detection system. And for the detection system, there is requirement of some technique. So basically for the viral detection, two types of system are used. One is the direct identification, other is the indirect identification. Because if you identify that this is SARS coronavirus 2, then only you will say that the person is having COVID-19. Otherwise, it can be a common cold, or it can be some flu, it can be bird flu, or it can be swine flu. Anything can be there, because the symptoms are somewhat similar. So on the basis of symptoms, we cannot identify. You require to have a clear-cut detection of this SARS coronavirus 2. So for this, the method is used. So in direct method, the genetic material or the protein sample which is present in the virus, it is detected. So you have to take the sample from the patient, and then you have to detect for the nucleic acid or the protein. So this direct detection system is right now used for this COVID-19 diagnosis or the SARS coronavirus detection, in which we here the genetic material is RNA. So the genome is RNA. You have to make the copy of that RNA, or you have to use some protein like spike protein. So you use that, and then you can design a technique. In indirect method, we use once the pathogen has get entry inside the body or the host, after that the organism or the host, it will develop some immunity like antibody. So once the antibody has been developed, after that the detection is not detecting the virus, rather it is detecting the antibody which has been developed due to the entry of that virus inside the host. So that is indirect method. So different types of methods are being developed. Okay, So right now, the detection system which is used for this COVID-19, you might be knowing about this, that the real-time PCR-based technique and the rapid detection kit, which is the antigen test, which is used. So this rapid detection kit is faster. You get the result in 20 minutes. For the real-time based, you require 4 hours. So if you see the basic principle behind this, the rapid detection kit, so there, this is a small kit which is actually a type of, we can say it is a type of blotting paper. So it is just a immunoblot type system where this device is actually called as the lateral flow device. So in this lateral flow device, there is a membrane inside this device. Okay, so I will use pointer. So this is a part where there is membrane. So this part is called as sample application port, and this is a membrane port where you can view the results. So when you put the sample in this pot, so like if you in this COVID-19 detection, we take the nasal swab or the throat swab, and then we dilute it in the some buffer solution, like we use the this virus transport medium or VTM. So in that you have to dilute it and then put a drop of this. So once you put that, so what so this is actually a absorbing pad, so it will absorb it. Then next to this there is a which is not been shown in this diagram, but if you go to the previous one, sorry. So here you can see that here this site is called as the conjugate pad. So in this conjugate pad there are labeled antibody. Along with that there are some conjugated gold nanoparticle, and this gold nanoparticle, what will happen, that if the virus sample is present, so the antigen which is present in the virus, like the RBD which is present in the spike protein, it will be recognized. So for that the antibody will be developed and they will be conjugated with the gold nanoparticle and they will be added in this conjugate pad. Now this virus antigen will be bounded by this antibody, and then it will go to the next part, that is this the membrane. So in this membrane it will flow, and in this membrane we have a capture of antibody in which we make a line of this antibody. Okay, so now what will happen here, there will be a sandwich type system where there will be antibody, over that there will be the antigen, and over that again there will be an antibody, and in that antibody there are gold nanoparticles. So if the virus is present, then this type of complex will be formed and you will get a clear line in this. So if it is positive, then you will see a line in this test, and this control is made just to detect that the system is working. And if you don't get any of the line, then it is an invalid system. And if you get only test and you do not get in this control, then it is also invalid. Okay, so next the detection system which is used, it is the RT-PCR. So here the sample is collected, that is the nasal swab or the throat swab, then it is added in the virus transfer media. Then from this the RNA is isolated, and after extraction of the RNA, the RNA is converted into cDNA, and that cDNA is used to check it in a real-time PCR. So in this real-time PCR, we do a quantitative PCR where we use different types of chemistry. So right now the chemistry which is used, it is the Tachman probe assay. So in this Tachman probe, there is a fluorescent and the quencher which is present in this probe. So this is a small oligonucleotide which is targeted to some type of gene, or you can target to some DNA sequence. So from this RNA you have made now DNA. So this is the DNA. Okay, so in this DNA, this what will happen is if the sequence is present, then this Tachman probe will bind. Now if the Stackman probe binds, now we have two primers. One is the forward primer and the reverse primer. And if these primers work here and they amplify this DNA and they reach to this site where the probe has bound, so what will happen? Then this probe, it will be detached from one end. So if it is detached, then what will happen? That this fluorescent and this quencher, they will be displaced. So when this fluorescent is dispatched, then it gives a signal to this real-time PCR, because there is a probe which detects fluorescence in this real-time PCR. So it will give a signal, and based on that signal, in each cycle, some signal will be generated, and based on that, the fluorescence will go on increasing, and based on that, you will get this type of curve, which is called as the CT curve. Okay, so here, the cycle where this is seen, so this is called as the CT value, and means the threshold value. And if according to the test, whatever threshold value has been decided, and if the result is at that level, then it is confirmed to be positive. And if it is not coming, this curve is not coming, then, or it is having a high CT value, then it is considered to be negative. So based on this, the detection is done for this COVID-19. Now, if this detection has been done and a patient has been confirmed that he is having COVID-19, now the next target will be to treat the patient. So mostly the people, they have mild cases. So in these mild cases, no drug is required, but there are some severe and critical cases where the treatment is required. And for this treatment, we require to have some antiviral drug or some other type of drug, which we'll be discussing. So in this antiviral drug, initially this chloroquine or the hydroxychloroquine, it was very much in the like the news that this is a drug which can be used for the COVID-19 patients. So actually this drug was used for the anti-malarial malaria. Then later on, some other antiviral drugs came, and this the drug that is the cancer drug, which was most effective, it was this remdesivir. And okay, so this remdesivir, it was also a effective drug which was given license to be used for this COVID treatment. So for using any drug, you have to target the replication cycle of this virus. So you have to, if you want to inhibit the growth of this virus, then you have to target where the virus is and how it is replicating. So for that, first is to bind to the receptor. Now for this, if you first neutralize this virus, then it will not be able. So this interferons and all, it can be used. So first target is to not allow the virus to be recognizing the receptor. So for that, first target is there. Then the second target is during the endocytosis. So during this endocytosis, this chloroquine was working, or hydroxychloroquine was working. Then next is the nucleotide or the nucleic acid release. After release, they will translate, and during this translation, you can stop that translation by using some of the antiviral drug like lopinavir, then ritonavir. Then the effective drug, that was the remdesivir, it is a base analog, so it just acts in the site where the genetic material of the virus is replicating. So here, this RNA-dependent RNA polymerase, the activity there, it is affected. So the viral genetic material is not allowed to replicate. So this was the targets where different types of drugs were used. So you can target to this all 9 sites, and any site drug can be made. So like this remdesivir, which was used as effective drug for this, it was targeting the replication of the genetic material. Then some more type of therapeutic ways were found where this is the use of this neutralizing the neutralizing antibodies for this coronavirus were used. So what are this? So you can use this monoclonal antibody. Okay, so monoclonal antibodies are antibodies which can be targeted to some antigens, and it can be manufactured in high scale, and they can be used because this virus, it attaches to the receptor of the host. Now, if you have an antibody which is made against this receptor binding domain, then what will happen? So if you have made for this, then what will happen, that the virus, it will not be able to bind to this receptor which is present in the host cell. In the similar type, there are some nanobodies also. So these nanobodies, they are some small sized peptides which act like the antibodies. So they are single monomeric variable. Like in antibody also, you will see that there is FAB region, which is the antigen binding domain, or where we see that there is a hypervariable region. So here this single monomorphic variation is seen in case of this nanobodies, and these nanobodies are very small antibodies which can be used. So they have just like a variable antibody domain. So this was also a type of therapeutic which can be used in this COVID-19. So which I have got from this publication that it was published in the Trends in Immunology. Then one more approach came, which was the plasma therapy. So what happens when the people, they are affected after the infection, they are cured, and once they have cured and they have survived, then you can draw the blood from this, because this blood will be having antibody against the virus, because the person has been cured from the virus infection. So certainly the antibody will be present in the blood of this individual who has survived from COVID-19. So you can use the blood, and then you can collect the plasma from this, and you can neutralize it. Virus neutralization has to be done, and then the serum has to be given to either a patient who has already been exposed with COVID-19, or to a patient who has not been exposed. So if you give it to a patient who has not been exposed, then if they are exposed, then what will happen, that there will be a virus neutralization, and due to that, the further replication of the virus will not occur. So this will be a type of like treatment which was also in the news, where you might have seen that the plasma therapy, but it was not that much effective later on, which was seen. Then in microbiology, we know about this probiotics, or there are some more nutraceuticals. So as we are discussing about the microbiology, so I will be discussing about the probiotics. So these probiotics, they are known to enhance, or they are known to be immunomodulators. So what they do, that they immunomodulate the hosts. So they can be given to the host, and then your immune system will be evoked, and due to the presence of this natural killer cell, interleukins and all, there will be virus clearance, and due to this, the effect of this COVID-19 will not emerge in the host. Okay, so this is also an approach where a number of clinical trials have been done for proving the probiotic effect on this COVID-19. So where a patient who has been affected with COVID-19, the persons who are working with the COVID-19 patients, like the medical persons, doctor and all, so for them also this clinical trial was done. And these are some of the prophylactic, no, these are the therapeutic approaches which can be used for this COVID-19. And beside this, one of the most important thing, or which actually worked, was the prevention. So always it is told that prevention is better than the cure or the treatment. So for this COVID-19, government made a number of guidelines, and by following these all guidelines, we found that we were not affected with this COVID-19. Even after being we saw that a number of cases went on increasing. The cases of this COVID-19 went on increasing. So certainly in the population, a lot of people were affected. Even after coming in contact with that people, we were not affected due to this preventive measures where we maintained social distancing, washed our hand, used face masks. No, here one science is required. Why to use this face mask? No, the guideline was given that you should use multiple layered face masks. So a study which has been published in ACS Nano, it says that aerosol filtration efficiency of common fabric used in respiratory cloth mask, it was studied and it was found that if we use multiple layer of cotton, silk, then chiffon, cotton with flannel, then more than 80% of the particles which are below the size of 300 nanometers, they were filtered out, means they didn't enter. Okay, so the virus is approximately the COVID-19 or the SARS coronavirus. It is only 100 nanometer approximately. So certainly if there it is, then certainly 80% of the virus cannot enter through this because of the mechanical filtration. So when the aerosol is coming, that time also if you have used a multiple layer, due to the mechanical filtration and the electrostatic filtration, a very less percentage of the particles below 300 nanometer can enter. So due to this, we can say that this mask, they are very much effective. Okay. Now, we talked about what is virus, how it replicates, how we can prevent its replication by use of different antiviral drugs. Now, we also saw that the preventive measures which we used, they were also effective. Now, the most awaited thing which was there, because even if we are exposed or we are using mask, till what time we are going to use this mask, or till what time we are going to use this all preventive measures. So we require to have something which will be effective to get us rid of this COVID-19. So for that, this prophylaxis is there. So what is prophylaxis? Prophylaxis is preparedness of ourselves for fighting against this any type of pathogenic organisms, like here for the COVID-19. So this is the vaccine. So what are vaccines? So if you see, then the vaccine race in development of vaccine for this COVID-19, it was very much fast, and a number of candidates came, and a very rapid this preclinical trials and clinical trials, it came out, and we had developed the vaccine. Now, if you see actually how these vaccines do work, then when the virus gets inside the host, then inside the host also there is a defense mechanism, which is the immune system of us. So in this immune system, there is the adaptive immunity, which is developed whenever we are having some type of infective agent inside our body. So here the infective agent, it get entry, and after the entry, our host defense mechanism, it responds, and the proteins or the antigens which are present over the surface of this virus, they are being recognized, they are presented, and we get antibody against that by our immune system. And that antibody, it can go and it can neutralize the further, if the virus is encountered, that can be recognized and it will be neutralized. So this the B cells, which are the antibody producing cell, in this B cells, there are antibody producing cells which are called as the plasma cells, and there are some cells which are being kept as memory, which are called as the memory B cells. So this memory B cell, it recognizes the same antigen or the same pathogen. If it is encountered for the next time, it will be recognized by this memory B cells, and if the same pathogen comes, then the antibody producing cell will replicate and they will make antibody. So the response, which is the secondary response, it will be very quick. So this, the theory behind this antibody production or the memory cell, it can be also mimicked. So here it is due to the infection. So in the similar way, if we give this some of the part of this virus to our immune system, and our immune system is activated and it is having developed this antibody producing cell or the memory cell, then it will be a type of immunity which we can develop. So the same approach is used when we develop the vaccines. So here the vaccine development for this, normally it takes a lot time, but in case of the COVID-19, it was given permission, and the different phases, it were very much fast, and the approval of this vaccine, it took only 1 year for the development, whereas if you see, just for approval, it takes more than 1 year for any regular vaccine. So what are the different approaches which were used for this coronavirus or the COVID-19 vaccine development? So you can have a different type of vaccine. So different approaches are there, like you can inactivate the virus. You can have some parts of this virus. You can have a live killed virus, which is the inactivated virus. You can have attenuated virus. You can have a vector. You can have some unit. You can use DNA as a vaccine. So different approaches are there for which the different type of mechanism is used. So first we will talk about the weakened virus. So in this weakened virus, already the virus which is present, it has been weakened. So how it is weakened? So we just do some type of culture in this, some type of mutation we will do, such that the virus is there, but the virus is not able to cause the disease. So there is immunogenicity, but there is no pathogenicity. So the pathogenicity of the virus, it is weakened. Okay, then the other thing is inactivation. So in inactivation, what we do, we kill it. So how we can kill it? So we can kill it by heat treatment or by some chemical treatment. So in that way, what will happen? This virus will be inactivated. Now you can use either of this, weakened or the inactivated virus. Then you can inject it to a patient in form of vaccine, and the immune system will be evolved and you will get an immune response. So this was the first approach. The second approach is the viral vector vaccine. So here, normally the virus, it is having a vector. So you can use a vector as the mimic to mimic the virus system. So what will happen? This virus will, the virus which you have made as a vector, this will mimic the virus, but it will not replicate. So it is two types of these vectors are there. One is the replicating viral vector, and the other one is non-replicating viral vector. So for this, we use some weakened type of virus, like for replicating viral vaccines, this weakened measles is used, and for non-replicating, such as adenovirus is used. So here, what you will do, a small part of the genome of this coronavirus, it will be added to this vector. Now, it will be a type of recombinant vector, which can be easily delivered to the host system. So inside this host system, it is not going to cause disease because it is having just a small part of the viral genome, which is for some type of gene, like the spike gene. So if you put the gene for this spike and then you put it and the immune response is generated, then what will happen? As this virus will go inside the host cell, then inside the host cell, this part of the gene will be expressed and the protein will be made, and that protein can show the immune response. So in that way, this vector vaccine can act. Then the next target was to use nucleic acid. So you can use the RNA or DNA as the vaccine. So there you will use the messenger RNA. So just RNA. So directly the RNA or DNA, they can be used by injecting them, but there is one more process by which it can be done. It is encapsulation of this DNA or RNA inside a lipid coat. So that is called as the lipid nanoparticle. So this lipid nanoparticle technique is there, where typically some isotonic or isotable like lipids are used, and then we make a type of this type of encapsulated uh, liposomes, inside which you can put the plasmid DNA or the messenger RNA which you want to use as a vaccine, and then you just deliver it. So once so because these are liposomes, so they can easily fuse with the host cell and they can get inside by the endosome formation, and inside this endosome it will be released. So here, as this is a messenger RNA, it will go inside the protein synthesis system of the host, and inside the system the protein will be expressed and you will get the antibody against that. So the immune system will recognize this one. So in the same way, this plasmid DNA can also be used. Okay, so these were the different approaches by which the different type of vaccines were focused, and a lot of work was done to find out effective vaccine within a year. According to the WHO data, total of 170 vaccines were given permission for the preclinical tests, and further after preclinical test, 63 confirmed and they were used as the clinic during the clinical trials. And after the clinical trials, like if you see the different candidates which were used, they were protein subunit. One more thing was there, protein subunit is also there. I think I forgot it to tell you. So you can take some part of the proteins, which is called as the protein-based vaccine, in which we use some subunits of the protein which is present in the viral structure, like the spike protein, and make it use it as a vaccine, and then you can get immunity against that particular antigenic molecule, that is the protein. So these all approaches were used, and different types of, like you can see that whatever the candidate vaccines. So in this protein subunit there were 19, then viral vector there was 10, for DNA it was 8, for inactivated virus 9. So likewise there is a number of different type of vaccines which were the platforms which were used for the development of the vaccine, and they reached up to the clinical phases. And if you see, the effective candidate was Pfizer, then Oxford, then Bharat Biotech. So there are a number of the vaccine candidates which reached up to phase 3, and after the phase 3 trials, some of them were confirmed. Okay, so it started with the preclinical trial, then is reached to the phase 1, then so in the clinical trial there is phase 1, then there is phase 2, and there is phase 3. So many of the candidates, they were just disqualified, and finally very recently only 2 vaccines in India have got approval for use. Okay, so that 2 vaccines are the Covishields and Covaxin. So this Covishield is being developed by the Serum Institute. So, and this Covaxin will be developed by Bharat Biotech, and the rate and the cost of this all has been decided, and according to the yesterday's news, a lot of doses of this Covishield has been ordered, or it has been, the order has been given, and the vaccination rounds have started in India. So this Serum Institute developed this, is going to develop this Covishield, and this Bharat Biotech is going to manufacture this Covaxin. What are the like the ways, means the, we can say, platform where this vaccine are working? So this Covaxin is inactivated vaccine, and this Covishield is a non-replicating viral vector. And the efficacy, it is more effective. This Covishield is showing more effective, and the data for this Covaxin is not present. And if you see the storage temperature, that is very much important, because in India we don't have the proper storage system at end user level. So here this both vaccine, they can be kept at 2 to 8 degree, which is normally possible in keeping them in ice cool pack, or you can just keep it in a refrigerator. So there it is easy to maintain. But for protein subunit or DNA vaccine, you require to have deep freezers, which is not that much feasible in case of India. So for that, this was that maybe it will be the one of the reason that it was, these vaccines have been given the permission to be used in India. And after the use of this all vaccine, we will be in a position where there will be no threat of this COVID-19. Then everybody there is chances that the total population will be not vaccinated, but huge population or the major population who are in the threat of this COVID, they will be immunized by using the vaccine, and the rest people, they will develop the herd immunity, which is told. So here, what will be the next? The next will be that there will be 3 types of people. One will be the person who has not been infected. Okay. The second person is who has been infected. The third person is who has been immunized. Now, the person who has not been immunized or who has not developed the immunity due to infection, they are in threat. But due to the herd immunity, this infected person, they can have this immunity, and the vaccinated person will certainly have the immunity. Now, to find out who was infected, who is not infected, who has been vaccinated, a surveillance is required. Then one more thing is there, that as we have as the origin of this virus suggests that it has come from the animals. So similar way, maybe the animals will also be affected with this. So this surveillance is also required to find out whether this virus has gone to the animals and it has done some type of mutations in the animals, and it may also, again, there is chance that after some mutation it can again come and it can become an epidemic. So this all concerns are there. For that, sero monitoring and sero surveillance is required, which is going on, and the government is doing all this thing. So this all thing is there for the COVID-19, and I will thank you all for listening to this. So we have to keep calm and just have to say bye-bye to Corona, so that very soon we will not have any threat for this COVID-19. So we should have a positive attitude, and by the this all whatever things I discussed, this all approaches will certainly be a way to get rid of this COVID-19. Thank you. Hello.
Thank you so much, sir, for a very, very, very enlightening and informative session. Deepika?
I think it was not that much boring, so I had tried to make it very simple.
Not at all, sir, not at all. So students, please ask questions if you have any queries.
Excuse me,
I
sir.
think the lecture was self-explanatory. So if you have any query, then you please ask.
Sir, I wanted to ask that since you mentioned in your presentation that in the starting there were about 154 plus vaccines for COVID. So at the end you said that there are only 2 vaccines that have been approved. So what were the tests that were undergone? Like what all tests did it undergo to ensure that these 2 vaccines are safe?
Okay, these 2 vaccines are safe. So during the development of these vaccines, like I can show you that usually what happens that there are different phases of clinical trials which are done. So there is a preclinical stage where in vitro tests and all are done. Then after that there is phase 1 where few peoples are taken. Then there is phase 2 where hundreds of volunteers are given the dose. Then comes the phase 3 where thousands of volunteers are given the vaccine or the dose of the drug, whatever it is. And finally, after the approval of this phase 3 only, the authority give approval for manufacturing and the use of a particular drug or vaccine. So in case of this COVID-19, it was very much fast because it took very less time. Like if you see, just I can show you one thing that this is not new that the people they started. So if you see this Moderna and Pfizer who use this messenger RNA based vaccine, they started this development of this vaccine just after the first sequence of this Wuhan virus was it was uploaded. That time they had started the work for developing this mRNA based vaccine because the mRNA sequence was known. Now based on that sequence you can target any gene like the spike protein, which is very much required. If you have a vaccine against that spike protein, then you will be having an antibody which will directly go and bind to that spike protein, and in that way the virus will be neutralized. And so this all approaches they took different phases, and finally after the approval of third phase where thousands of volunteers are given the dose of a particular this vaccine, then only this was confirmed. And beside that there is one more problem. Like if you see, this Pfizer was approved in US, but in case of India this was not approved because this messenger RNA, there are some type of logistic problems also, which I told you that the temperature problem is there. So in the rural area, this vaccine cannot be sent because you require to keep them in ultra low temperature, which is not possible at the interior places. But if the vaccine is it can work in 2 to 8 degrees, then it is very easy. You keep it in like ice pack, then you can transport it. You can keep it in a general refrigerator also. So many things are taken in consideration, and after the successful trial of the phase 3, the vaccines are approved.
Okay, thank you so much, sir.
So regarding this, if you want elaborated like analysis, you can just go to the WHO site and you can search for this landscape of novel coronavirus candidate vaccines development worldwide. So in that they have given the whole data. It is like it is Excel file. So from that Excel file, the summary part I have taken. So in that Excel file, everything has been given, which has been published in 8th of January 21. And these
Okay.
two vaccine which I had discussed, it is only from India, because in India, this two has been given approval. In other countries, some other vaccines are also there. But in India, this two has been given approval. So I was discussing that.
Yes, Deepika, you have some questions from the YouTube.
Yes, ma'am.
Yes.
There's a question from Aditya Rudra. Only 2 vaccines have been approved, considering both are mRNA based. Also considering that Covishield didn't undergo phase 3 completely, they have been approved. Is it safe for mass immunization?
That part I cannot answer, because I am not the right person to answer that, because I am not a medical person. So, or even I am not involved in development of drugs or vaccine. Just with the knowledge of my microbiology and the studies, I have explained this one, but that thing I cannot explain. Because in India, one more thing is there, that feasibility is also very much important to be used. So maybe the decision makers have taken the feasibility also, because if it is very much costly also, then it cannot be afforded by the government. So if you see the Covishield, it is negotiated at around 200 per dose, which can be at least we can say it can be afforded by the government, or individual level also it can be afforded. But if it is like 20,000 per dose, then maybe it is not feasible. So maybe that will be the concern.
Okay, sir. I think that's all. Thank you so much, sir. It was a very informative session. We're looking forward for a lot of collaboration with you. Thank you so much.
Thank you, ma'am.
Thank you, sir.
Thank
Dr.
Thank you,
Pratik, please take over.
I will stop. Hello.
Ji, sir?
Hello. Yeah, Bandana ma'am. Oh, actually, one of our lab mate was there who was student of you, Nancy Awasthi.
Oh, yes.
I had met, she told that just tell hello to ma'am. I was a student of her. So we were lab mate during doing PhD at NDRI.
Okay.
So she was master student that time.
Yeah, okay. Thank you so much, sir. And say my hi to her too. Thank you, sir.
Thank you, ma'am.
Thank you, sir.
I think Gurpreet, you can start.
Deepika, can you please share your screen?
Yes, ma'am. Doing it. Just a minute.
Good evening, sir. I think we have Dr. Ashok with us now.
Hello, you can hear me?
Shall we start?
Can you
sir?
hear me?
Yeah,
Yes,
yes, sir.
Can you hear me?
Yes, sir, we can hear you.
Yes, sir. Good
Oh,
evening,
good.
sir.
Good evening.
Good evening. Good evening. So I need to share my screen. Screen.
Sir, I would like everyone to know about you, sir. A little introduction of yours.
Oh, sure, yeah.
So, good evening, everyone. It's a great pleasure for me to introduce our next speaker, Dr. Ashok Kumar. I think I need a complete session to introduce him, but after squeezing the information to the maximum, I would like to share some of his achievements and accomplishments. Dr. Kumar did his BSc Medical from DAV College, Chandigarh, MSc Microbiology from Kurukshetra University, Haryana, and PhD in Medical Microbiology from the Postgraduate Institute of Medical Education and Research, Chandigarh, in the year 2003. Later in 2003, Dr. Kumar went to the US for postdoctoral training.
In 2004, he moved to Kresge Eye Institute of Wayne State University and continued his postdoctoral training there, where he got promoted to a research scientist position in 2006 and later in 2010 to assistant professor. Currently, he is a tenured associate professor of ophthalmology, microbiology, and immunology at Wayne State University School of Medicine. In addition to this, he has also served as an associate dean of graduate school at university. Now, coming to his research accomplishments, Dr. Kumar is a nationally and internationally recognized expert in infection biology. The common thread running through all his scholarly activities is to study the host pathogen interactions in microbial infection. His research is largely concentrated on ways to enhance the host innate immunity to fight against infection. Indeed, his research on Zika virus in 2017 and more recently on COVID-19 in 2020 was highlighted among major accomplishments of Wayne State University, and that received a lot of media attention also. He's using cutting-edge and high-throughput omics technologies such as transcriptome and metabolomics to address various scientific queries. His laboratory is currently funded grant from National Institute of Health with a budget of more than $10 lakh per year. In addition, he has funding from various research foundations, pharmaceutical companies, along with several research collaborations. Regarding the publications, his publications till date include 70 original research papers, 3 review articles, and more than 120 presentations at national and international meetings. Awards and honors can never stay away from such eminent scientists. In 2011, he received RPB Special Scholar Award. In 2012 and 2018, he was recognized by the Wayne State University School of Medicine with the Faculty Research Excellence Award, a distinction that is given to a few faculties. In 2013, he was the co-recipient of Dr. Jack Ryan Research Award. He was also nominated for Postdoc Mentor of the Year in 2013. He has mentored more than 50 trainees, including undergraduate, masters, graduates, PhDs, postdoc fellows, medical student residents, and clinical fellows. Dr. Kumar has been frequently asked to serve US National Institute of Health and Department of Defense and numerous other national and international grant review panels. Dr. Kumar serves on editorial board of 6 journals and as a reviewer for more than 50 journals. In his illustrious career, he has served multiple distinguished committees. So everyone, please join me in welcoming our esteemed speaker, Dr. Ashok Kumar. Sir, please.
Oh, thank you, Gurpreet. That's such long introductions.
That was just a bit of what you have all achieved.
So how I share with I can share my screen with Deepak Malik. So I share it or so share my screen.
Yes, so please go ahead.
Oh, how do I'm not actually, I'm more familiar with the Zoom than this one. So I say, how do I share it? Sorry.
So
there's a bar at the bottom saying present now.
Okay. So it's so present now. Okay. And then I say.
Then you select your window or your entire screen that's more comfortable with you. In a window, you can select the presentation you want to share.
Oh, okay, presentation I want to share. So presentation is. So actually, I already opened on my screen. Let's see. Okay, share. Okay, got it.
Got it?
You can see my screen now.
Yes, sir.
Not yet. Okay. Now, yes, sir.
Yes, sir.
Yes, sir. It's there.
You can see it.
Yes, sir. Yes, sir.
All right, perfect.
Okay.
So I want to thank you. First, like to thank Sandeep. I know she find me on the LinkedIn. That's how we get connected, and that's why I want to encourage all of you to make those connections. And that's you can. I mean, internet is pretty wide open, and make those connections. You can find expert, and that's a wonderful way to connect with expertise and English is important for your education. It's also important for your career. So I'm really thankful to Sandeep for finding me and bring me on this Discord. I'm also an alumni of DAV College. So I still remember those memories, and there used to be a guy used to use the chole and kulche there. Every lunch break, you go there and enjoy those chole and kulche. Still vivid memories I have of Chandigarh. So it's a really pleasure. So what I want to share with you, I want to share the overall about COVID-19, starting to really give some historical perspective, what happens in terms of host pathogen interaction, and what are the current therapeutics available, and then we will finish off with the vaccine. And maybe if your time permits, then I'll probably share a couple of slides which show about our research which we are doing in our own lab. So let's, as a microbiologist, let's try to make some, there's a lot of confusion, right? Because it's a new disease sort of, and people are, all sort of information is coming in the media and research and everywhere. So I try to make it simple. First thing we need to understand is that COVID-19 is a disease, okay? It's not a virus. So there's a lot of, people use it interchangeably, right? So, and some people just say, oh, this is a positive COVID-19 test, regardless of disease or symptoms. Who named COVID-19 is the WHO, right? Who initially named it COVID-19 because at the time the virus was not known. So it got picked up by the press and media, and they start thinking COVID-19 is a virus, right? But you can see my whole screen, right? Or I'm doing a presentation mode.
Sir, it's half visible. I think you can go to the full screen.
Oh, full screen. Okay, sorry.
Yeah.
Okay, yeah. Let's see. Go back to full screen now. All right. Right.
Yeah.
So that's how really it gets started. Now, look at the headlines in early 2020. So this is, look at if you read the headline, it says SARS-CoV-19 can last few days on surface, new experiment findings, and study real how COVID-19 remain infectious on cardboard, metal, and plastic surfaces. This is pretty a nomenclature. It's wrong. COVID-19 is a disease. So I give an example of TB, right? Tuberculosis. Tuberculosis is a disease, but TB is caused by Mycobacterium tuberculosis, right? So just want to make sure this is clear. So is COVID-19 is a appropriate name for the disease? We can argue about it, and we can discuss about it. So what happens is most of the viral disease names, they do have the virus name which actually describe the disease what they cause. So for example, can we say COVID-19 virus? Yeah, it says it. Oh, what it means? It means coronavirus disease 2019 virus. Does it give you any information? Not. I mean, what disease? What kind of disease? It's a lung disease? It's a heart disease or kidney disease? There's no information there, right? So in contrast, when the SARS came, right, they define it a severe acute respiratory syndrome. Now we know that it has something to do with respiratory system, right? So instead of COVID-19, maybe they should have defined it as SARS-2, because we already know SARS has happened in 2003 and 2003, right? And 2012. So it would be better if they just call it SARS-2. But anyway, so the whole struggle continues when they started this naming. So everything started in a really wrong foot. So then first they call it 2019 nCoV-2, right? What does it mean is nCoV stands for a novel coronavirus. Again, this is very inconsistent when we name the virus. So it's pretty misleading actually. So when you say novel, means something has totally appeared, but that's not the case, right? It's probably the least novel virus among all respiratory disease-causing virus. We have a flu virus, right? There are a lot of viruses which can cause respiratory disease, but it's probably one of the most least novel virus. So again, 2019 nCoV is also a wrong name. So then what we actually have when all these emerging viruses comes, we have this International Committee on Taxonomy of Viruses, ICTV. So they actually named it SARS-CoV-2, and actually that's the official name. So right. So and the name of this virus, because why call it COVID-2? Because it's 96% identical to the SARS-CoV, which happened in 2003. So it makes perfect sense to call it COVID-2, right? So as a microbiologist, I encourage all of you to keep these two entities separate. COVID-19 is a disease. SARS-CoV-2 is the virus which caused COVID-19, right? So at least as a microbiologist, we can correct the people, right? And also, it's very important to understand when we are just saying the SARS-CoV-2, we can always refer, oh, there was something that's happened in 2003. So we got a lot of information way back, right? So that's how we have this a lot of data already available, 10 years worth of data. We can always connect these dots back, right? So these are some of those misnomer places. Now, in terms of the coronavirus are actually these, we call them a positive strand RNA viruses. This is how we classify the viruses. Positive strands, they have a DNA viruses, we have RNA viruses. I'm sure you must be learning in your a basic microbiology classes when we talk about the viruses. So this is a positive strand, means they have a positive strand, they just go into the cells, they immediately start making a second copy and immediately start making the transcription, and they make those proteins, and we will talk about those in little bit more details. So there are two type of viruses. We call them an alpha viruses and beta viruses. So these are the most common type of viruses, which are responsible causing the 10 to 30% of human infections. Among these, the RNA viruses, this is also a unique thing about the coronaviruses. What they did is this is one of the viruses, which is the COVID-19. It was isolated in 1960, and then it was also isolated. The same virus was isolated in 2001, and there is only difference of 2 amino acids. So these are pretty conserved. Their genome is not that crazy mutating as compared to the flu virus, right? And what are the other features about the coronavirus is bat, right? The bat is their natural host for these viruses. But they also have the ability to cross the species. So from bat, they need some intermediate host, and then only they can cause an infection in human. Usually they don't jump directly from bat to the human. If a bat bites you, it's very unlikely that they can just go and transmit a disease to. So they need some sort of intermediate host, and that's critical, right? So if you look at the history of the coronaviruses, so we had in 2003, that's the SARS, right? So it's actually originated from bat. Bat somehow bite this civet, and from civet it goes to human. Then in 2012, we have again still because bat is their natural host. They go to the camel, and somehow from camel it goes to the humans. This is how it is transmitted, and this is probably the most lethal type of MERS is more lethal than even COVID-19. Then we have the bat. Now this is a pangolin, and that's basically how it has been. That's been connected. It's originally started in China, and that's from pangolin it goes to human. So what I'm telling here is that you need an intermediate host to transmit this disease to human. So, but now each host actually they have their own viruses. So if you look at this list, these are the human coronaviruses, and they're usually called mild, flu, no stuffy nose and cold and the mild fever. But these are really mild respiratory infections. Pigs, they have their own coronaviruses. Camel have their own coronaviruses. Cat, human, horse, mouse, whale, chicken, sparrow. So all these animals and birds and all these species, they have their own coronaviruses, right? So it's not something that it has disappeared from another planet. We have always been exposed to them. They have always been circulating in different hosts, right? So just give you a little bit of historical perspectives. So the first human coronavirus, like sort of really was discovered really in 1930s, and it was called an infectious bronchitis virus (IBV). Then in 1940s there was a discovery of this a mouse hepatitis virus, and this is usually called it causes a gastroenteritis virus. But in human, is actually the 1960s the one there was these are the two human coronavirus H29 229E and OC43. These were discovered in 1960, and then we know the history, right? So then we have SARS-CoV 2003. Then there is another virus appeared in 2004. This is usually called a mild, mild to severe pneumonia in infants and adults. And then we have 2005 HKU. Then we have 2012. We have MERS-CoV, and 2019 we have SARS-CoV. So what it says is that these viruses are always been there. They keep coming, and it's just the severity and the nature of epidemic, it really varies. But COVID-2 is the one that really caused a pandemic. Most of the other ones, they really cause an epidemic. I mean, they're pretty localized, or they will contain very quickly. They're just very limited. But COVID-2 has caused a pandemic, and that's why it's in the news everywhere now, right? Now, so this is a very cool things which people do. What's called a molecular clock analysis. What they do is they take the virus, which has isolated maybe tomorrow, and then they do some sort of predictions. They go back to the genome sequencing, and they identify at how many years back this virus might have evolved. And what it really tells is that the SARS-CoV, this N63, which was actually discovered in 2004, but it has diverged from this 229, which was discovered in 1960, right? About 500 years ago. So this is like how they're, what we call them in molecular clocks, means they do a prediction that from at 500 years ago, they diverge, they become into two. They're still the coronaviruses, but they develop some other mutations and how their spectrum becomes different. So that means these sort of indicate that they have been for more than 500 years ago, these viruses are still around us. And what other thing? Next. Okay, so now the question really is now we know that the bat is their natural host. Then how does they can infect in humans, right? As I told you earlier, that they need an intermediate host. So what the first panel shows is when they took this COVID, the coronavirus from the bats, and infected individuals, they did their sequence analysis. They looked at their genomic RNA, and they found that they are closely matched. So what it indicates is that yes, the COVID-2 recovered from human actually came from bat, right? But we know that bat cannot infect directly human. So what is the intermediate? When they look at this pangolin, they took the virus, they did the genome sequencing. They found actually this also has a quite good similarity between both, which is recovered from bat and recovered from human. Okay, fine. So that make a connection, right? How we make the connections, where it came from. Now that does not prove. So what they did is they took this RNA make those proteins, right? So this is DNA, RNA make proteins. Is some of molecular biology, and then in the bat we have this. Like you can read this. These are the 5 amino acids, right? Total. But these 4 amino acids have been changed in those these have been changed in the virus which was recovered from human and as well as which was recovered from pangolin. So that makes another connection. Now the protein sequence from the pangolin virus, recovered from the pangolin or from human virus, they are similar, right? Where the genetic material was similar from bat to this one's. Okay, so now this making a connection. Yes, somehow this is now making a more confirmatory that it actually came from pangolin, right? So is that a sufficient evidence? So the scientists go further and they found actually now we will talk about this one. So there's a binding domain and there's another protein which is required for the virus to enter into the cell. And that protein is cleaved by the hosts. For the host, they will cut the protein, and by that, the virus get internalized into the cells. And they found that the virus recovered from human are the only one which actually have that specific cleavage, where a host cell will cut the cleavage in the virus, and then only it can enter, right? So it's from the RNA sequence to the protein sequence to all these mutations. So that really concludes basically yes, indeed, somehow the virus go from bat to pangolin, and from pangolin it goes to humans. So that's how people study the transmission of the viruses, where it came from. There's a significant amount of research being done about these exotic viruses, right? They can found in different environments, how people can really make. So we need to be ready, because and it's not the end. There will be more viruses will be coming down the line. Now, in terms of the structure, why we call it coronaviruses? So the corona stands for the crown, right? This big crown. So crowns are actually indicated by these big spikes here. So they have these spherical structures, and that's why the name is coming from. So now there are the 4 proteins which is expressed on the surface of the virus, and one of them is called the spike protein. This is the S protein, which is responsible for making these crown type structures. And this is a trimer. There's 3 proteins come together to make this spike protein, right? As you can see, 3. Then we also have a membrane proteins, and we have these the annular proteins E. So these are and then inside we call them a nucleocapsid protein is called L proteins. Now, when we talk about the vaccines, right? So vaccines are it's the best way to develop vaccine is to develop the vaccine against the surface proteins, because they are expressed on the cell surface. Now, in case so there is most of all these vaccines are actually targeting a spike protein, the S protein. This is about they're targeting this specific protein on the virus, right? But for the diagnostic purposes, most of the diagnostic purposes using as a PCR, they actually target a N protein, which is inside the virus, and this nucleocapsid protein is which is wrapped the RNA of the viruses, right? So this is genetic material. So we also in our own lab use a PCR to detect these nucleocapsid proteins, and so for the vaccine development we use the surface proteins. For diagnostic purposes using RNA, we use the N proteins, or there are other proteins, right? And these are those other proteins. So I told you that there are 4 structural proteins, which is essential for virus to make the structure, and that includes spike protein, annular protein, membrane protein, and nucleocapsid proteins. But inside when they get into the cells, they make more proteins, like another 20, 25, 16 of other proteins. We call them an NSP. These are non-structural proteins, right? So these proteins, what they do is that their function is really to help the virus to replicate, to get into the cell, to replicate, and to exit out of the cells. That's really their real function. So if we are talking about the targeting the drugs, so we can target the drugs towards the structural proteins. So the vaccine will be towards structural proteins. But if you're talking about the antiviral drugs, so antiviral drugs, we need to target against these proteins, right? Because these are the ones which help the virus to replicate inside the cell, and we can block any of these proteins to have a antiviral effect, right? So just give an overview, simple view. I mean, this is more complicated than it visualized. So what happens is, so the virus actually go and bind the cells on the receptor. We will talk about this called ACE2 receptor, which is I'm sure you heard in the news. Then virus can internalize into these endosomes. They degrade it, and then they release their RNA. And this is a positive strand RNA, so it will make its negative strand. And this positive negative strand, they replicate, right? They just keep replicating. Now we have the RNA, then the ribosomes will come. The ribosome will bind, take this RNA, and then they will start making these proteins, right? So, and that it happens into the ER, endoplasmic reticulums. Then they get packaged. So all these proteins, then this genetic material go into the packaging. We call them the packaging industry, and then they get packaged, then they are released, and the cells is lysed. So when we're talking about the therapy or the drugs, so we can block any of these pathways. So there are the drugs. They can inhibit the binding of the virus to the cells. They can inhibit the entry. They can inhibit the viral applications. They can inhibit the assembly. They can inhibit the viral release. So we have learned a good deal about these different therapeutic targets, and that's where the big research is happening in terms of the antivirals. And a lot of knowledge we can actually gain. We can gain from other viruses which we already know. This is how a typical virus will replicate. So now, in terms of how do we identify that what is the receptor which is required for the virus to entry? So in the SARS 2003, the SARS-CoV is already known that it actually take a receptor called ACE2. So it was very easy. So we asked this question, does the virus use the same receptor? And yes, indeed they do. So what they did is that you take a cell, human cell lines, you express a mouse ACE2 receptor, you express a civet. This is those intermediate hosts, right? The civet, swine, or bat, or human. So this is a cell. These cells are expressing this receptor ACE2. Then you put the virus on the cells. So virus is a color as red, and wherever you see a merging, means red and the red and green mix, it become yellow, right? That's we call them co-localization. So if you see over here, when you use a mouse ACE2, they don't get infected. That means you cannot use a mouse to study the SARS-CoV infections. But if you use any other the ACE2 from the civet, swine, bat, and human, they get infected. So this proves that the viruses need ACE2 to bind and to get into the cells, right? Now, is the ACE2 now the next question comes is, oh yes, we now know there is this receptor, but which cell type in human body or tissue express this receptor, right? Yeah, obviously it's known that that it's lung disease, so they look at the lung, and yes, indeed, and this is a 2004 paper. Yeah, they abundantly normal lung tissue express this ACE2 receptor. Even the fibrotic lung express this receptor, and then there are a couple of cell lines we use in experimental studies to replicate the virus. And they also express these ACE2 receptors. But the ACE2 receptor is not only expressed into the in the lung, but actually it has found that this is also expressed in the oral mucosa, in your buccal cavity. They have this expressed ACE2 receptor. Even our intestine, they have ACE2 receptor. These are the specific cell type in the GI tract. We call them enterocytes. They also express ACE2 receptors. So small intestine, large intestine, they also express the receptors. And that will make a perfect sense now. It's not only the lung. Yes, people die because of the lung infections, but the patient who has COVID-19, they experience diarrhea, they have nausea, the vomiting, they lose the sense because it's expressed on your in the oral cavity. You lose the taste, right? You lose the senses. And why? Because they express the receptor. The virus can infect your oral mucosa, right? So one of the tests which is they're doing saliva. So usually with nasopharyngeal swab, they put a cotton swab in your nose and take this out and put PCR. Now we also have a test called a saliva. You spit. You develop a saliva. You put in a tube. You can do PCR, and by doing the saliva, you can actually tell whether you are positive for SARS, whether you carry SARS-CoV-2 or not, right? So this is the reason why the saliva has also become a very popular method for diagnosis, because we express ACE2 in our mouth. Now you must be wondering now. Oh yes, we have identified the ACE2 receptor. We know this is how I get into the cell. So why don't we block it, right? Just find something to block the receptor so the virus cannot enter into the cell. But the reality is actually ACE2 is a very important protein, a receptor, and it does normal functions. It's not only yes, virus use it, but they have all these proteins have a specific reason why they're expressed in those cell types. And this is typically the ACE2 receptor works. So there's I will not go in more detail. This is more of a physiology and biochemistry, but the idea is, so there's we call a RAS system, angiotensin renin system, where they make this ACE2, and ACE2 basically is actually has a very important role in tissue protections, right? This is how they protect the tissue. So they're really protecting mechanisms. So the virus is really clever because it's used that receptor, and that's how it hijack the system of ACE2 signaling pathways, and instead of becoming protection, it becomes now that reminder, right? So what can we do to block to this? So certainly we cannot block ACE2, but what we can block is we can block the interaction of this spike protein with ACE2, right? So and that's how this again, like I told you, the spike protein is a candidate for all these vaccines already came in market. There are drugs which can specifically block the receptor interaction between spike protein and ACE2. And there's another clever idea which is I think still is in a development, and this is called a soluble ACE2. So we have a normal ACE2 expressed in our cells. What if we synthesize a soluble ACE2 and we inject in the patient COVID-19? Now the virus will bind those circulating ACE2, and it will not cause a problem because your tissue ACE2 is still being expressed. So then again, this is what we call decoy. You basically fooling the virus, and so that they can just bind it, and they will not bind the host this express on the cell receptors. So that again, this is all in the clinical, preclinical stage. We'll see how this approach will pan out. The idea is to stop these interactions. Now, the other approach. So certainly we cannot target ACE2, right? We can only block the interactions. So there is another approach, and that approach which I told you early on, that the virus which was recovered from these COVID-19 patients has a cleavage site which is only present in the virus from this human host. And what happens is that virus, when they bind the cell receptor, they can bind, but they need another protein called furin. So that protein will cut, it will chop a protein on the virus, and that basically is activate, and that virus can internalize into the cells. So that will be a clever idea. Could be, even if the virus has bound to the ACE2 receptor, can we block the activity of this specific protein? So the virus it can bind, but it will not be activated, right? And in fact, these are drugs. So these are the different mechanisms. So binding is one part, but then for all viruses, they need to be activated by using this specific protein expressed on the host, right? So this is one of the drugs. This is called a TMPRSS2 protein. So again, I told you the virus can bind with ACE2, no problem, but then this needs to be activated. Now there's a drug called a nafamostat, and this has been already in clinical use. So you can actually use that drug, you can block the activity of this. Now, if you block the activity of this specific protein, now the virus cannot internalize into the cell. This is really the cool idea. And what scientists have shown that this is a graph. So if you see this normal cell, we are looking at the entry of the virus into the cell. Okay. Now, if you use this drug, the camostat drug, and this is a different concentration, right? We have tested with MERS, SARS, and SARS-CoV-2 against all these viruses. Now, if you increase the concentration, you see a reduction in the entry of the virus. That means this drug can be used. And I'm not sure, I think this is still in a clinical trial, whether can we use this drug to stop the entry of the virus, right? So now, what are the antiviral drugs? There are, like I told you in the beginning, there are many steps which can be targeted. The one is a blockage of binding of this is the binding. We can block the binding of the virus to the receptor. These are the drugs, and we will talk about more. The monoclonal antibodies also target the same area. We will talk. I have one slide to share with you. Now, once the virus get into the cells, then they start making these translational, right? So these are the drugs. And the fluoroquinone has also been um, extensively, people have championed for it, and it has become so much controversial. So where does the fluoroquinone act? The fluoroquinone actually acts when the virus get internalized into the endosomes, and then they acidify the endosomes, and then the virus cannot be released, right? That's where the fluoroquinone is acting. But then there are drugs. So all these drugs are having a different effects. The most popular drug is called remdesivir. So remdesivir is a drug which actually block the RNA polymerase, RNA-dependent RNA polymerase. So it's basically acting at this stage, right? So once the virus RNA get into the cells, they need to replicate. So this drug block the replication of those, the transcription of those viral RNA, and this drug has been, now developed in the US, and still being used. And there are other drugs. They have different functions. So this is the area what we call them an antiviral drug discovery to combat COVID-19. And so this is the drug remdesivir by Gilead. And what this drug has, no, this is an old study, but there's a new study came up. They actually found that if you so it only prevent even it's really shorten your hospital stay. So if you, for example, if you have been sick, if you don't take this drug, maybe you are out maybe on 15 days of hospitalized. But if you take this drug, maybe you will out of the hospital in 11 days, which is still good, right? So, but it's only prevented. It's really good when you can diagnose them early on, but it's not going to prevent you from, if you are really a severe case of COVID-19, it will not prevent that. So it will delay. It will, prevent your hospital stays, but it's still good. It can be used in combination with other drugs, right? So in my lab, actually, we started this is 2017, and we've been working with Zika virus before we started looking at the COVID-19. So I'm sure you heard that Zika virus also caused an epidemic, right, in 2016-17 in Brazil, where they found the pregnant mom. They're delivering these babies, and these babies actually have a smaller head. That's called microcephaly, because the virus is from the placenta of the mom, it goes into the fetus, and that's where they actually go into the brain of the developing baby to kill those brain cells, and that's why you have a smaller brain, you have a smaller head. So we did studying the pathogenesis of the Zika virus. But so these are some differences, right, between COVID-2 and Zika virus. But there is one common between these two viruses is that so both of these are actually the post-strand RNA viruses, right? That means they need a similar host machinery to calculate. So the reason why I'm showing you here is because we can get a lot of information, what we learn from other viruses, and we can apply that knowledge to the newly developed, to a new pandemic or newly discovered viruses. So science is continuously evolving, right? So this is one of the data which we already published this using with Zika virus. So this is a drug. This is metformin, and I'm sure people who have diabetes, they use metformin a lot, right? Because this is anti-diabetic drug. So we actually found this is the green cells are the cells which has a lot of viral infection. We are using Zika. This is another compound, and you can see here that this drug actually pretty much block the viral replication is drastically reduced, right? So we are now testing whether can we use this drug which we discovered against Zika virus. Can this also be used for SARS-CoV-2? That's one of the project. I will not go into more details. So we also have a couple of other drug target disease, another compound which we discovered. We've been working on looking at Zika virus infection. Again, these are the green cells that are infected with Zika. This is one drug. They sort of almost drastically reduce viral replication in these cells. There's a drug that's called benzamyl. So the virus is actually low cholesterol. If you give a lot of cholesterol compared to this one, the cells keep a lot of virus will replicate. And this is a drug called benzamyl. we actually discovered that these drugs can pretty much regulate cholesterol influx, and you can use these drugs as a to inhibit the viral application, right? Now about the chloroquine, actually this is my 2005 paper. I published this paper in 2005. Where actually we did show that when you use this is one of the we use a viral infection as indicator, right? You put the cells, you put this, this polyas is a viral mimic, and when you increase the concentration of chloroquine, you see reduction in the antiviral response. This is just an idea that's what we have been showing. We actually have, even before this COVID came, we have already been working with this the chloroquine. And what I'm showing here is these are the top is the cells. Again, when you put the cells with chloroquine, you see a significant reduction in the viral replications. And we have also tested the chloroquine in animal models, right? So this is a mouse eye. I will show a picture of how the mouse. Well, this is right now the mouse. When you give a Zika infection, these mouse eyes have a lot of lesions in them, and when we give them dose, just give them, they're systemically give these mice, and we can say there's significant reduction in the pathogenesis. So what it really tells is that the concept of using chloroquine is not new. I mean, it has been, people have published, long back, and even we have showed the data. What does it really works in COVID? That's we don't have. I don't think we really have a good clinical data. Now, in terms of how do we study a COVID-2? So we can study a Zika virus. Our lab also studied chikungunya. We also study dengue virus. So these viruses are they are not that lethal. So we can use we call it, in microbiology, we learn that called biosafety levels 2 pathogens, bacteria, fungus, all of these are BSL-2, but SARS-CoV-2 is a BSL-3 facilities. So I have this colleague, Dr. Arunanga Swami, so we work with him at UCLA, and so we have a culture system. We also have animal models, and this is how a SARS-CoV-2 infected cells will look like. And again, I can show you an example. In cell culture, yes, it works beautifully. Chloroquine works, right? Does it really work in patients? No, it's not that great. We don't have good data, right? So, but there are drugs. This is how we do a screening of the drugs which can inhibit the virus in culture cells. From there, then you need to do an animal study. From there, you do a big animal study. Then you go in human. That's how the drug is discovered. So, but anyway, the whole thing about the chloroquine is, yes, the chloroquine is we call it a research drug because it does a lot of things. It inhibits the viral applications. It also has an anti-inflammatory property. So a lot of things. So we certainly there's a lot of things. So again, it's really inclusive. There are still, I think, a couple of clinical trials going on, but it's still very inconclusive. We don't know if it's good or bad. So in terms of the pathology of the COVID-19, so this is how a normal lung will look like. So you can think of the lung alveoli as a balloon, right? So in Holy, we just we fill the water in balloon, and at a certain capacity they can hold it, but after a certain time they will rupture. That is exactly what happens in case of the lung of these bronchi of these COVID-19 patients. So the virus, we inhale the virus through nose or mouth, then get into your lungs, and that's where it activates the immune system, right? T cells and macrophages gas comes in, and this is a very early phase. But as the disease progresses, this lung alveoli get filled with the fluid, right? And that's why we want it to be very clean, because you want an air. Only air has to be in the lung alveoli. They expand, they contract, and air exchange happens. But if the balloon gets filled with water, then you know what's going to happen. It's going to rupture, right? And then it will cause a massive inflammation, and that's why the lung gets collapsed, right? And then these people, they cannot breathe. And this is all because of these inflammations and getting the fluid filled with these lungs. So I will not go in more details, because typically this is a scenario what happens in case of you get a nasal or oral, you acquire the virus, it goes into your lungs, it activates your immune systems, and which is very immune system is important to do protection, but then the immune system fails. There's too much inflammation going on, and that leads to what we call the cytokine storm. Like we call them farm, right? So somebody's roof will be blown, or house will be blown because of this is now be stored in your body of all these cytokines and immune system going on. So the virus from the lung has to go into your heart, go into your kidney because it's your systemic viremia, right? And they all have these receptors. They have more virus being produced. The virus, then they have more systemic inflammation. Again, that leads to what we call cytokine storm, and we have a multiple organ failure. Right? It started with lung, but now we have multiple organ failure. Similarly, in lung, as I told you earlier, the virus hijacked this ACE2 system, which is very important for protection. Then this system gets dysfunction. You have a vascular permeability. A lot of immune cells are now coming to the lungs, and we have a pulmonary edema, and then we have a respiratory syndrome, and that's how people die. But if you have really a wild viremia, you survive and you become asymptomatic. And this is really what we call the cytokine storm. So I told you the drugs, which is antiviral. Now there are the drugs which can really try to make it quiet the cytokine storm. So because the cytokine storm is the one that this is because of most of these patients die because of the cytokine storms, right? So this is, I just put it in a simple way. So we have a stage 1 where you are asymptomatic, and we have an innate immune response, which is very protective actually for the host. Then we have this sort of window when you can go this way or you can go that, right? So this is called a non-severe stage. And then we have a severe stage, that means now your immune system it was good, but now you have a cytokine storm, your immune system is dysfunctional, you are having multiple organ failure, and that's how that leads to the death, right? So we really have this window of opportunity in the stage 2, where we can give a lot of intervention, antimicrobials and anti-inflammatory, so that the patient doesn't go from stage 1 to stage 3, right? And are there drugs? Yes, there are a lot of drugs actually which have been used. These are in clinical use for other inflammatory disease, like when you say IL-1 blockade is called anakinra as a name of drug. There are drugs which can block IL-1, IL-6 receptor blockage. There are the drugs which inhibit these are pathways which are responsible for inflammations. So you can use those drugs which can try to fight your immune system, right? So they don't go crazy. And then the other part of this, the equation is that to use this, they also recommend you to use an antibiotic. So now your immune system is already compromised. You already have viral infection. Then you might actually get a secondary infection from bacteria. So all these drugs, the anti-inflammatory drugs, they also recommend that they need to be combined with broad spectrum antibiotics just to avoid a co-infection by bacteria, right? Because then you have virus infection, then on top you have bacteria, the chances of the mortalities are very, very high. What are the other? So we talk about the antiviral. We talk about these immunomodulations in reducing the inflammation. What are the other therapeutics we have in pipeline? What we call an antibody or plasma therapy. So plasma therapy is pretty popular. So what happens if you're young, you get an infection, you get recovered. So that's what they're asking you. If you were recovered from them, donate your blood, then they take the plasma, then they can use it for prophylaxis. So this is usually for prophylaxis, means they can be given to those doctors or the nurses who are on the front line taking care of patients. Or if you're critical, if you're ill, then they can give to you. So that what it does is that it really neutralizes the virus, right? That's also a principle of the vaccine, because now you have the antibodies against the virus because you recovered. You can donate it. They can be used. We call it a plasma therapy. But the drawback of this, when you have pandemic situations, you have so many people getting infected, and now it's really difficult to have a sufficient amount of the serum, and you need multiple doses, right? Because it's like blood. You give 1 package and 5 packages of the blood. The same thing, you need a lot of plasma therapy. So what can we do? So then to remove these drawbacks, then you can use a monoclonal antibody. Monoclonal antibodies are similar, but you develop them in the lab. So you develop these monoclonal antibodies specific to those the surface protein of the virus. You develop in the lab, you make a drug, you give those patients, same IV, and they can go circulate and they will neutralize the virus. That's another approach. And our president Actually, when he got sick, he was admitted. He was given this one of the monoclonal antibody. Now this monoclonal antibody is developed by Regeneron, now is in market and can be used. But they will be effective if you diagnose them early, and if you have a very low viremia, if you have too much virus, there's no way you cannot, it won't be that much effective. Again, the early diagnosis, and then you are like I told you, you're in that stage 2. At that stage, probably it's going to work. Now, in terms of the vaccine, I think the previous speakers already talk about those the clinical trials. So I'm not going to tell you the clinical trials. Rather, I give you what are the underlying principles of all these vaccines, what are the different platforms. So what is a vaccine? So vaccine is they help us to develop immunity, right? To prevent, to limit the infection. So basically, you are sort of mimicking an infection scenario in your body, but you don't get sick by taking these vaccines, right? So you are sort of training your immune system, right? So your physical effect. Okay, you do gym, you do all those exercises, try to make your body protected. That's really the principle of using these vaccines to enhance your immunity against the specific pathogens. And we have been getting this vaccine, when we're born and in different stages of our life. Now, just to give you a simplified view, again, just coming back to the targeting these vaccines. So we have the virus, they enter into the cell using this receptor, and within the cells, then they start making these, they replicate and they make these viral proteins, and this is how the virus is assembled, and it's released, it will kill the cells. So at the molecular levels, so what this virus is, when this virus is picked up, what we call them antigen presenting cells, these are the immune cells. They're circulating in our blood all the time. They are our army of they are looking for any evasion in our body. So they just picked up. They take this virus, they chop it. They chop this, make the smoothie, and then they basically presenting all these different proteins of the virus on the cell surface. Now this cell is now labeled. They say, oh, I'm infected. I need help. So now then we have the first cell which is coming, we call them a T helper. So these are T cell, B cell, right? You will learn in the immunology. So that T helper cell will come, they recognize those receptors, those proteins, viral protein on the immune cells. Now they get activated. And that was when they get activated, they activate the B cells. B cells are the one which make those antibodies, right? And they also activate what we call the cytotoxic T cells. These are the cells which will go and they will kill the infected cells. So it's a multi-pronged approach. It's just not, so the vaccine is actually are the one who are responsible to activate this multiple immune system of the body. Now, the other important aspect of the vaccines is that there are some vaccines, polio vaccine, you just get one time in your life, done with it, right? So there's no need to get it again. So the idea for those ones is so that these vaccines can lead to development what we call the memory T and B cells. We have memory cells, right? So if I get infection today, then if I get infection after 1 year, I still have the memory, and I can quickly evolve the immune response. That's called memory, right? So it's also important that these vaccines can develop these memories. So again, this is a little more details. So again, there are the these are the three immune cell types, which is very critical for vaccine development. It's also critical for the antiviral immunity, what we call them B cells. Again, B cells are the one with they can differentiate, they become plasma cell. That's how they make the antibodies, right? Now, T helper cell, these are they are helpers, that they are the first one to come to see an infected cell which has engulfed the virus, and then they get activated. They make these cytokines, chemokines. They activate the macrophage. They induce inflammation. They also help in the development of proliferation of the B cells. So the idea is just to amplify the immune response. Means there's the invasions, right? So you have one army on the border, and then say, we can see some signal there. We need help, and then all the other soldiers will come to play to contain that that invader, right? Then we have a CD8 cytotoxic cells. So CD8 cells. Now these cells are also circulating in the system, and when they see an infected cells, what they do, they just kill the cell. They don't care. They just kill their own. They kill the host cell because they recognize that this specific cell is infected, and we want to kill it. If you kill the cells which has a virus, then the virus cannot replicate, right? So these are the different three arms of the immune system. They need to be activated. The good vaccines are those which can activate all these three arms of the immune system, right? So what are those vaccines in development? So their platform could vary. So this could be just a virus-based, and I'll tell you briefly about all these. This could be a viral vector-based, nucleic acid-based, this could be protein-based, but the concept is pretty similar, which is I told you the activation of these immune cells and having those memory. So let's take the example of the first one, what is called the virus-based vaccines. So in virus-based vaccines, we have you take for example, we take SARS-CoV-2, and there are two types. One is called a live attenuated virus and vaccine, and another was an inactivated virus, right? So what is live attenuated virus? So these live attenuated viruses, you take the virus, you pass it through them a series of animal cell, human cell, animal cell, human cell. Basically, you're putting them different. Like I showed you, we do a cell culture-based model. You take from a human, they put the virus in the animal, from animal to human. So you need a lot of passaging, and hoping that by when the virus passes from different cell types, they pick up some mutations which will reduce its ability to cause a disease. That's called a live attenuated vaccines. And this is a Dr. Vincent at Columbia University. What he's showing is on his wall in the office. These are the plates. This is we call tissue culture plates. And so you need a lot of plates. It's really a long process. It takes many years for the virus to get its mutations and to identify which virus has a specific mutations. Then you can amplify this virus and you can use that virus and then you can inject it, right? Now these viruses, they will not cause the disease, but they will evoke the immune response, right? And what are the examples? The example of the live attenuated virus are the smallpox, chickenpox, yellow fever virus. These are examples of the live virus or MMR. Now the other type of virus-based vaccines are what we call them inactivated virus. These are very popular, right? So it means, so you take the virus, you make a tons of big container of the virus, and you use some chemicals. You use a formaldehyde, you use heat, you use a UV light, and you basically you're inactivating the virus, but they can still keep their ability to activate the immune system, right? And this is called inactivated. So what are the examples are? Hepatitis, flu virus, polio virus, rabies. These are the inactivated virus. And I think the one which is being developed by Serum Institute of India is belong to this category. It's an inactivated virus. That's what they will be. But the idea is same. When you inject in patients, they replicating live they will be replicate, you generate. These viruses will not replicate, but they will just activate those immune cells. Now, what are the other one? Other type. Other one is we call them viral vector vaccine-based vaccine beans. So here, you take. I told you those. The virus has those spike proteins. Then you take the sequence of the spike protein, you clone into a plasmid in a vector, and then you use a viral vector. These these viruses. They don't cause. You're just taking the. You're basically renting somebody's house, right? So you take this envelope of the viruses, but you're putting the gene of a different virus, which is which you want to induce immune response. So that's really the concept. Now these replicating viruses, when they go into the cells, they will replicate. They will not cause disease, but they will express the specific gene which you put in those viruses. Then they will start making the proteins against that. You are using that DNA or RNA, and then they will start making those proteins. Again, the protein will be expressed on the cell surface. That is how immune cells will be able to recognize. And so these are pretty safe. I mean, they provoke very strong immunity. But the only problem with this one is that for example, we might have a pre-existing immunity to a specific vector. So it's very important to choose what kind of the envelope you're going to choose, what kind of viruses capsid you're going to choose, right? So for example, we might have an immunity against the some of the adenoviruses we have immunity, because if you're using those ones, then you put in the body, they get neutralized because I already have immunity against those the surface proteins, right? So so it's very important to pick up the good house basically you are putting there, right? What is an example of this one? This is a newly approved Ebola virus belong to this category. We call it replicating, or they took the gene of the Ebola virus, put in this vector, and then now they can be used, right? What the other type is, what we call them is non-replicating. So we are replicating and non-replicating. You just simply put this virus will not replicate. It's get into the cells, start making those. You have gene like spike protein gene here. It go into the cell. It will start making the spike protein in the cells, right? And what is the example? AstraZeneca is one of those examples, which is I think is already in the market approved from UK. So AstraZeneca is the one which is using this non-replicating virus-based approach, and they're using a chimpanzee adenoviruses because chimpanzee is close to human, so they're using the capsid of the virus particle they're using is from the chimpanzee adenoviruses, and so this for the non-replicating we really don't have much licensing, but they actually AstraZeneca is using the replicating model for the vaccines, right? So what are the other viruses? What are the other approaches? What do we call them? Protein-based. Now, instead of putting the gene in the cells, why don't I put the protein? I can make the tons of spike protein and then give the people injection. Then they will develop an antibody against the spike protein, right? That's why we call it protein-based approach. Right now, there are about 30 teams in the US. They are working on the receptor binding domain of the spike protein, and that can be purified. Then they can be injected, right? Then the other one is what we call them a virus-like particle approach, means you make these proteins, right? And now you want to express those proteins on the cell surface. So you take a mimic, a small bubble, and what the approach will be, take those and try to mimic it as just like it behave like a virus, and this spike protein is being expressed on this fake virus, right? In fake bubble, droplets or nanoparticles, right? This is the nanoparticle-based approach where you're actually putting these proteins on the cell surface. Now they will be picked up again by the immune cells, they present into the antigen-presenting cells, and they generate the immune response, right? And there are 5 teams who are actually working on these ones. But again, the thing with these ones is that they're difficult to manufacture because some of the configuration, there are a lot of things technically goes under this approach, and even for the protein-based vaccines, you need a booster. Sometimes they need a more dose, multiple dose, and then you need an adjuvant, right? Now, what the most popular now? The most popular approach now, what we call them nucleic acid-based vaccines. So these are 2 types of vaccine. One is called DNA vaccines, and one is called RNA vaccines, right? So the DNA vaccines are the one you take the DNA of the spike proteins, plasmid, and you do a electroporation. So this is what we typically do in our labs. We take the cells and we culture them, and directly putting the DNA in your cells. Now this DNA will make the RNA, right? Because this is a DNA with RNA. The mRNA will make proteins, and this protein will be presented on the cells. Immune response is generated, right? And now, but to consider this, there is a lot of things need to be considered. What is a promoter? What type of promoter? Which kind of cells the human body can pick up this DNA fragments? Adjuvant, what kind of backbone you're going to use. So there's a lot of background work goes to develop these vaccines. The most popular and which is easily relatively faster approach is called RNA vaccines. So what it is it really bypasses the DNA, right? We have a DNA go RNA and then the protein. So you say, why don't we just inject the RNA itself? That's it. So you're going to bypass this, making the DNA to RNA, put the RNA, they start making protein, boom, done, right? So and this is really this is the pros using these RNA vaccines is that for both RNA and DNA, this is pretty safe actually. They're very easy to develop, and this is a really what you are doing really, you're making a genetic material in the lab, and you can purify them. There are a lot of high quality, you can make those genetic material. That's it. You can manipulate it. You can put some, now we have this new what's called the new variant is coming up. No big deal. Just take this take the new just change the nucleotide sequence to specific that protein. You can do, I don't know, you can just done within a couple of months. We have now, a new variant, no big deal, right? You can just modify the DNA. This is all can be done, but all other approach which I told you earlier takes a very long time. Now, one of the drawbacks is that we don't know the long-term efficacy, right? We have this data about they are efficacious, but we don't know the long-term effect, right? So these are the two most popular, which is being used in the US, Moderna and Pfizer. In fact, I got a Pfizer vaccine just 8 days ago. I'm waiting for my second dose. Will be January 25th. So these are two vaccines which are developed in the US. They have 97% efficacious, and they are really, all these are based on the RNA vaccines, RNA. This is another sort of the pipeline, but I just want to share. So these are the current scenario what we have. So we have a AstraZeneca, which is a viral, which I told you, viral vector. We are using a chimpanzee virus and chimpanzee viral vector, putting the DNA, and then they will release. And this has already been included in India, Britain, and 3 countries. And it's expected by 2021 they should be producing about 3 billion doses. And this is probably the cheapest too. We have the Pfizer mRNA-based vaccines. This has been approved in Britain, UK, USA, and 20 other countries. And this is supposed to be, this is a really tiny amount, but it was expected by 2021 they're going to produce 1.4 billion doses of these vaccines. And Moderna is available in the US, also been approved in Canada, Europe, and Israel. And I think I did not follow much about nothing, but India is also developing but this is an inactivated. So we talk all about different approaches, right? Viral based, mRNA based. These are viral based, inactivated, mRNA based. So these are different platforms. These are the 4 popular way you can develop vaccines. But the fastest way to develop is the RNA based vaccine, because you bypass the DNA steps. So it's really incredible, and this is really something we as a scientist, I feel proud about it. It's such an incredible journey of this vaccine development in the US, especially by the Moderna, right? So in December 1 is the first COVID-19 illness was documented, December 1st. Now all these different phases it has gone through. By December 11, we already have vaccine started in the US. So it's literally in a 1 year period. And this is so incredible. Now, the question is, yes, we have the vaccine, but what do we do till we get a vaccine, right? So there are I showed you 3 approaches. I showed you the antiviral. I used some information about antiviral, anti-inflammatory, those drugs. So those are actually very good because they actually help you, at least when you don't have, you can use those drugs. And this graph really tells is that if you can reduce, so we have a 5 days once you acquire the virus, 5 days you stay, you're not contagious. Means you get infected, but maybe not that contagious, right? And then you're 9.5 days when you become contagious, means you can spread the virus to others. So, and what it really tells you, if you reduce by just half a day of this contagious phase, you can actually, drastically reduce the cost. Hospitalization, can be drastically reduced, and cases. And same thing is we reduce by 1 day. This is the cost we can reduce. And if you do reduce by 6 days, so how can we really achieve this? Minimize this contagious phase, right? To make it shorter and shorter. And of course, the best thing is prevention is that we all know that, right? Prevention is better than cure. So only thing is, yes, vaccines are coming in. It may take some time, but we should still keep our prevention. Using those masks, and social distancing are important. But during this phase, it's important that there is the antiviral, plasma therapy, monoclonal antibodies, anti-inflammatory. So these are all the drugs which have been used in other diseases. They can still be very good drugs during this phase, right? So now I have spent maybe just like a couple of slides. So these are a couple of things we need to mention. How we diagnose if you have COVID-19, right? So there are really two approaches. One we call a PCR-based or molecular method-based approach. And you take the nasal swab, so you take the what's called the saliva, and you run a PCR test. And what is the positive PCR test means the person is actively shedding the virus, so it's contagious, right? And negative PCR test means that the person is not shedding the virus. It's not contagious, right? So how do you, if you are negative, our CDC, the Center for Disease Control, which is the governing body in the US, it's maybe like an ICMR. So they recommend that if you have two negative PCR tests done 24 hours apart, then this person could be called as a non-contagious, right? So then the other test, we call them ELISA or serology. And what they do is they're detecting an IgM, IgG, right? So it's just like a pregnancy test, they're going to test, they have a strip there, we can positive, negative, IgM positive. So what it does is, if you, in the strip, you have a negative for IgM and IgG, that means there's no recent exposure to COVID-2. If you're only positive for IgM, it will tell that you are likely you're at early stages of infections. If you are positive for both IgM and IgG, that means you're in the middle stage of infections. And if you're only positive for IgG, means either you have recovered the disease phase, or you are really at the last stage. So now the question you may ask me, then which test we should use, right? And the fact is that we cannot, we need both, right? They are different purposes. So the serology will test you whether you have been exposed. The PCR will test whether you're contagious. So we cannot replace any of those ones, right? So we need both. And the serology is also important when we do a serosurveillance. How many people have been, having this antibody, or how many people have been vaccinated. You can randomly draw blood and look for IgG and IgM for SARS-CoV-2, right? And this is a simple principle of IgM, IgG, and these are antibody, right? I told you the vaccines, the vaccine approach. So if you get an infection you get infected by SARS-CoV-2, and the first your antibody will be IgM, which is quickly comes, within 3 weeks, and then it fades out. Then you have IgG. So that's why you need to detect IgM and IgG ratio that will tell you whether you are which stage of the infection you are. If you are both IgM, you are in the middle stage. If you have more IgG, you are at the late stage. If you have more IgM, then you are early stages of infection, right? And these are like I told you. So there's this company Select in the US has this one, and that's basically sort of mimic like a pregnancy test, is positive, negative, and some people are using a lot of these techniques. So there are many of these companies are actually developing these for diagnostic tests because that's important. But on the other hand, there are drugs, there are companies and selling left and right, have no sensitivity, not much, the specificity. So sensitivity, specificity is critical, when we study microbiology, right? And what are the sample type? You are using saliva, using nasopharyngeal swab. Do you need a special instrument for a PCR? You need a PCR machine. Then it also depends if you are in a village or a PGI. They are all, do they have those facilities to run those tests, right? It's really a combination of multiple factorial which play key roles. So now I'm going to spend maybe another 2-3 minutes to show what we are doing in our own lab. So we just reported that the patient who died of COVID-19. So in US, I think it's everywhere because there are the eye banks. They take they take their eyes and they can use to transplant the cornea, and the people who are blind, right? This is an organ transplant. They can donate their eyes. So what we found is that there are a lot of people are dying because of COVID-19. So we want to ensure that that these patients who die of COVID-19, they don't they don't harbor viral in their eyes, because if you take a COVID-19 patient and take their organs and you transplant into a host, then there are the chances that you may end up those recipient get infected by COVID-19. Right? That was our initial rationale. And what we found is that we analyzed about 66, 70 of those donors. Some of them, they exclusively died because of COVID-19. Some died because of other reasons. So we did a PSA testing on those eyes, and we found about 13 to 12 to 13% of these patients who died COVID-19, they do actually have a viral RNA in their cornea. So this is structure of the eye. The front eye, you see the that's called cornea. The cornea has epithelium, stroma. And what we are showing here is that we can actually use an antibody against the spike protein. We can stain those tissues. And we actually found some of those patients, they do actually have a presence of, at least an indication that the virus was there. Whether it's live or not, we don't know. But it indicates, so this is one of the way to exclude those samples. Then we just say, oh, don't take these samples because this came out to be positive. They cannot be used for transplant, right? So the other reason why we study the eye is very important. Is if you think about it, we know this specialty called ENT, right? Ear, nose, and throat. Then we go to these specialties. So the eyes are actually connected to the nose. They are connected to the nose. We call them it's called a lacrimal duct and a nasal lacrimal duct. So they open up into your nose here. Now, if a patient has a COVID-19, has a virus in their nose or throat, then there's potential that it can go to the eyes, so it can go all the way around. So it means you get. So this is so we are trying to understand: can the eyes become can the virus be transmitted through the eye? That's what we actually study. So what we have this cartoon developed by one of the postdoctoral fellow, Sneha Singh, is beautiful. What it's showing is that even if you have a mask, you protecting your nose, you protecting your mouth, but if your eyes are open, if somebody coughs or sneezes on you, then those droplets can go into your eyes, right? And through this connection through the eye, they can go to your nose, they can go to your throat. And we're trying to understand whether it can be transmitted. That's really the underlying theme of our research. So what I tell students is that I don't know how many of you are using the contact lenses. So if you're using a contact lenses, maybe it's a good idea if you can avoid during this pandemic, just use your regular glasses. At least you have glasses. If somebody cough on you, you cough on your glasses, you're not going in your contact lenses, right? So this is one of the ideas we are, the Sneha, she's the fellow, and she's really the spearheading these efforts in our lab. And we are having system. We have now a mouse, and we actually putting we just topically put the drop on the eyes, and when we want to see whether these mouse they develop these severe lung infection, and by doing that we can prove that yes, indeed, we don't have the evidence yet. So, and then we testing what are the other cell types in the eye can be prone to. They can pick up virus. They express the ACE2 receptor. They can transmit. That's what we sort of study. So these are the people in my lab who have contributed to all this work. And so Nikki and Xavier they are now in this big group. So and if you see here, actually, I'm surrounded by these things. Singh is a very powerful name, right? So these are very talented. These PhDs and the postdoctoral fellow. They are really the who works in the lab, and they amazing scientists. So I really encourage all of you, if anybody is looking into the career in research, it's amazing. It's an amazing. And one of the things I tell students is that I can guarantee you that this is the only job I can speak. So this is the only job in the world where you are getting paid to do what you want to do, right? So there are all other jobs you actually have a certain protocol, but this is something you truly enjoy, enjoy doing that, right? And I think I can just leave it here. Also, before I go, so I also need to mention that how many of guys, how many of you driving the American car? So actually, this is our city called Detroit. This is where our lab is. And our city is called a motor city because we make all the cars. So Ford, GM, Chrysler, all these cars actually are made in Detroit. So it's a big car company. And I And I always make jokes. Anybody who joined my lab got a free car. Right?
Wow.
so this is so this is what I am putting here. I know that I put this cartoon here. I know in 2020 we were actually we want to flatten the curve, but now we have the vaccine. We really want to steepen the curve. So I really, when the vaccine comes, I really want all of you to take the vaccine. And what I will do is I think that's it.
Okay.
I can stop sharing the screen.
Thank you very much, sir, for an interesting and informative session, and also for gracing your important work and sharing with us your findings today. So there are some queries from student side. So can we have a small question answer session?
Yeah, of course. Yeah, no problem.
Okay. Dr. Deepika, unmute yourself, please. Unmute.
Good evening, sir. There's a question from Anil Sahu that can we consider the upcoming vaccine is 100% validate or it needs some improvement? And will it be helpful for new strains, means the mutated ones?
So can you repeat the question again?
Yes, sir. Yes, sir. So can we consider the upcoming vaccine is 100% validate or it needs some improvement? And will it be helpful for new strains, means the mutated ones?
Oh, yes, sure. Yeah, so this is a great question. So right now, the efficacy of these vaccines which is developed in the US, I know more about US ones, it's about 95, 97%, right? Means they're pretty efficacious, right? Now, does it need to be improvement? I think, I mean, there will always be an improvement. It's an ongoing process, right? So I think what they're looking at, even I get vaccinated, they're tracking me, they're tracking all those things. So hopefully when we have more vaccine, individuals get vaccinated, now we have a bigger data base, and then we can have more, then only we can tell how efficacious it really is, because initially they did like about 40,000 to 60,000 volunteers, they did the vaccinations. So definitely we need the long-term data, and that only can tell how efficacious these vaccines are. Now, in terms of the new variant, so Moderna has already done their testing, and Pfizer has already done their testings. And remember I told you, when they get vaccinated, you develop an antibodies in you, right? You take those serum of those vaccinated individuals, and you do what we call the neutralization assay. Means take those serum, can those the serum can neutralize the new virus, and they have tested, and they both of them they claim that their vaccine has a can develop an antibody which can also neutralize the new variant. So what is the new variant? There is a mutation, I mean, one or two amino acids. I don't know exactly what one. And I know one of the examples. We need a three proteins, right? Three trimeric proteins. So this could be a mutation, but that does not really change much of the structure unless it's really a dramatic structures. That probably will then they may need to develop new vaccines, or another variant of those vaccines. But I will not worry about those new variants. I know what the best example. My feature does not change. My face does not change, no matter how much makeup I put. Right? These are my features will be the same. So these are certain features will remain the same. You can have I may put a glasses, I may put different haircut. But so what of these vaccines are developed is really looking at the three-dimensional structure of those the spike proteins. So, and even if something happens, they can quickly develop. I told you, RNA vaccines are very easy, and they can quickly develop. Like they did it in 1 year. Now they have technology; they can do maybe a month.
Okay, sir. Thank you so much, sir. And there's another question, sir, from Amanjot, that what are the best vectors to be used for DNA vaccines?
What are the best vaccines used for the DNA vaccine? So for the DNA vaccine, I think most of those they are using the adenoviruses, because what the reason is, like I told, for the virus to enter into the cell, you need a receptor, and most of the human cells actually pretty much have the receptors for the entry of those adenoviruses. So AAV9, AAV5, the different variety of those adenoviruses, and that's what most of the DNA vaccines are being used using the adenoviruses.
Okay, so do we have any other questions from the audience?
Yes, ma'am.
Yes, please go ahead.
So I have a question that do we Indians really need a vaccine? Because without vaccine, India is amongst the top countries with highest recovery rate. And the number of deaths caused due to COVID is nearly the same as deaths that happen due to cardiovascular and respiratory diseases and also accidental deaths. The only problem was that we did not have enough space in the hospital. So if we had enough space, then this coronavirus might not be a problem for us.
So the question is, you're saying that, let me try to reframe the question. So you're saying that we have more people dying because of heart diseases and all other complications compared to corona. Is that correct? Is that a correct or then is that a correct statement, or I'm trying to get what the real question was, or is we don't have enough vaccine, or we don't need to worry about too much about corona? So I'm sorry, I did not really capture the question. So yeah, the thing is, and this has been a sort of a politically or same thing in the US as well. People always say, oh, more people die because of accident, and more people die because of other diseases. But really the data what speaks, right? So you have why so many people have acquired the virus, and people are dying. The severity could vary, right? So I will not go in that direction saying, oh, more people die. Why do we care about corona?
Sir,
Okay,
there
so
is
there's another
another.
question from Mansi
Yeah.
Sharma. Can
Yeah.
you just share some light on that? Why do the viruses try to get into living cells? The students want to know this thing.
Oh, why the virus? Okay, sure. So virus actually, it's pretty interesting. So for microbial, like we have bacteria, virus, fungi, right? So virus cannot survive outside, right? So these are the intracellular. They need to get into the cell, because virus, they have what they have. I told you, like they have 4 proteins on the outside, then they have genetic materials. So just to make their progeny, to make more virus, they need to get into the cell, because they use the host ribosome, they use all the machinery in the cells, and then only they can replicate. Otherwise, if you put in the virus outside, they will die. And we don't even call them a virions. These are one particles. They cannot do anything unless they get into the cells. So they need it for their progeny.
Okay, sir. Sir Sandeep ma'am, you were asking something?
No, I just want to ask students that if they have any questions in the Google Meet.
Rufika wanted to ask something. Yes, Rufika,
Yeah.
please.
Ma'am, I want to ask, is natural immunity better than acquired immunity?
Is a natural immunity is better than acquired immunity? I would say that's pretty. I would say no, not really. So this is actually one of the myths is that if I already has been exposed to COVID, do I need a vaccine? The answer is yes, because when we do an acquired immunity means like vaccination, right? They have really optimized the conditions. Like I told you, the 3 arms need to be activated. You need to have those CD4 cells, CD8 cells, and memory cells. All these things need to be activated, and it takes really a long effort for the scientists to optimize those conditions. So by vaccination definitely gives you a more protections. And yes, of course, you'll be exposed to, naturally we've been exposed to certain we do acquire natural immunity, but I don't think that's a which is better? I think that definitely vaccine is better for sure, because they have been optimized to. They've done tons of data. They've done all these experimentals, and you also want to make it more universal. So even people from India, from different countries, and older people, younger people, African Americans, Indians, Caucasians, so all races and genders, all things need to be considered when they design the vaccine.
Thank you, sir.
Sure.
So I had a question.
Yeah.
There are many other diseases and other pathogens that are way more contagious than coronavirus, and their death rate is also very high. Why is it that coronavirus has posed such a great challenge to the whole world?
Yeah, so this is a great question. So one of them was, and one of the big example is Ebola virus, right? If you get an Ebola virus, 24, 40 hours, you're dead. It's very, very deadly, right? So, but the thing is, it's really the transmissibility is the what has triggered, right? If you're respirating, because you're transmitting, and this is one of the reasons why, cough, you sneeze. So that's what really has expanded. All those like Ebola, you don't need all these respiratory viruses have that capability, right? If I have any other viruses, you need a mucus, you need a direct contact with the body, you need something by any other means. You are transmitting this virus, but only the respiratory viruses are the one which has caused this pandemic, and they always will be the one which probably will not cause more pandemic, because you are transmitting at a very high rate. And on one hand, this is the one part. So if there's an epidemic, you can be easily control. So the Ebola was actually happened in Africa. Immediately the US sent their things and they really contained it, right? And now in Wuhan, they try to contain it. But it's just like now it's global, right? I visit India, I acquire some disease and bring it to the US, and that's boom, let's go everywhere. So the why becoming so pandemic because of its ability to transmit quickly and faster. That's one of the reasons why this corona has become so pandemic.
Okay, thank you, sir.
So there is a question by Sahajpreet. Is it necessary for someone who has already had the COVID infection and has recovered to get the vaccine?
Yes, I will recommend. And actually, let me see if I can still see. I actually have one of the. Let me share one slide. I think there are a lot of myths. Let's see, there's my slide. If I can share the slide, I think I can just put it. That's good for everyone to. Okay, so I think I forgot again. Actually. So I need to do share the screen. How do I share? Okay, say present now. Window. Sure, let's see. So can you see this one? Everyone can see my screen?
No, sir.
No, sir, it's not visible.
Oh, so it's visible. Yeah, it's
Yeah.
now visible.
So this is actually our Handiford hospital.
Yes, sir, it's now.
You can see it, right? Okay.
Yes, sir.
So these are the 10 myths about COVID-19 vaccines. This is our actually hospital called Handiford Hospital in our local service. Actually, looked in the morning. I know this question is going to come. So the first question is, the vaccines are safe because they were developed quickly. These are not safe. This is also false, right? So like I told you, that's the reason they've been developed so quickly, because we already have a platform. They've been working for more than 10 years. That's so quickly, and this is RNA-based vaccines. The next myth is the vaccine can lead to long-term effect. This is also false. So there's no, yes, you can have some allergic reactions. But otherwise, there's no, it's very unlikely you can have a long-term effect. And I think that's you can get a COVID-19 from the vaccine, which is not true at all. Right? You don't get a disease from the COVID-19. So I really encourage you to, I can send you the link of this website, very informative. So now this is the question. I already have COVID-19. Do I need to get vaccinated? I don't need to get vaccinated, which is also false. And this is the CDC guidelines. Yes, the preliminary evidence actually stated that the vaccine offer better protections than having the virus. I think one of the questions I asked was about whether the natural acquired immunity is better than the natural immunity is better than the acquired immunity. So that the CDC recommend yes because it's much. These are way better vaccines are better. The other thing is people with underlying conditions like if you have diabetes, heart disease, are you? I mean, shouldn't not be vaccinated, which is also false, right? They are the one who need to be vaccinated the most because disease and these things. People with suppressed immune response should not get vaccinated, which is also false. The cancer people with cancer and autoimmunity, actually they are the one who need to be given the vaccine the most, right? And I think one of the most common, I think people always ask this, okay, this question is comes very often, if I'm pregnant or I'm breastfeeding, do I need to get, I mean, I should not get vaccinated, which is also not true, right? So you definitely need to be get vaccinated even if you're pregnant or breastfeeding. Now, the most important myth is about if once I get vaccinated, right? If I get vaccinated, I don't have to wear a mask or practice social distancing. This is also false, right? Because we say it is 95% efficacy because there are 5% of people who won't be protected. So this is never 100%. And you are going outside, you don't know, right? So which which one is vaccinated, which one is not vaccinated, right? So these are really some of those the myths about the vaccine. I mean, and I don't blame any of those because there's a lot of misinformation has been pointed by people. But I hope I clarify some of those. But I will be happy to send the link.
Sure, sir. So there's one more question that why there is low deaths in Asian countries as compared to others?
Say again?
So why there is low deaths in Asian countries as compared to others?
Oh, good question. So actually, I was reading just 2 days ago. I mean, that's pretty interesting. We have not done that experiment. Um, there are several ideas about the Asian, what we call them near the equator, right? So people in those hot zones or tropical areas, and there's one of the study actually found that people with India's and all those all over the equator, there's a big role of called vitamin D. So vitamin D, we have very high vitamin D levels. That's we don't know the really the is that because there is scientific evidence that actually people with COVID-19 they have this quite drop in their vitamin D levels. So who have been exposed to sunlight, they actually do have the vitamin D. Now, does that mean the people from Africa will not have those COVID-19, which is not the case, because when you're dark skin, you're really dark. Actually, even in the US, we have dark people, African Americans, they're actually more prone to COVID-19. And the reason is because they're not able to make enough of vitamin D because of the dark skins. So we really don't know the reason, but this is one of the theories. There are multiple theories, and one of the theories I was reading on one of the talk was about regarding the vitamin D.
Okay, thank you, sir. This also clarifies the question of Mannat that what is the role of vitamin D in COVID-19? And sir, there's
Oh,
another
okay.
question from SK Das that is increase the immunity against COVID virus? Does zinc increase the immunity against COVID virus?
How to increase?
No. Does zinc increase the immunity against COVID virus?
Oh, you mean
Zinc, sir. Zinc.
the vitamin D?
Zinc.
Oh, zinc. Oh,
Yes, sir.
I
Yes,
don't
sir.
know. I mean, I don't know the answer about the zinc.
Okay.
I haven't paid attention to it. Yeah. So sorry, I
Okay,
don't
sir.
know.
Okay.
Yeah.
No, it's okay, sir. So, do you have any other queries?
Yes, ma'am.
Okay. Yes.
So we were encouraged to boost our immunity by taking some vitamin C tablets and uh, drinking turmeric milk and all that. But once
Mhm.
we get vaccinated, common people are surely gonna leave this all of the sudden. Will it affect our natural immunity? Will it weaken it?
So once you are vaccinated, you think so. Because you are vaccinated, then people will go back to their. They will not do these taking turmerics and all. So remember that those turmerics and all, yes, they are they are sort of having anti-inflammatory, and we have the herbal medicines, tons of things, and sometimes we don't have those really evidence, but everything which you think make you immunity, right? It helps. So, but you got a vaccination and then you stop. Why would you stop? Because you still you can still drink the green tea or still take the curcumin or haldi and all those steps. I mean, why would you leave? But I think yes, but I put into the diet point of view, yes, it's very important that diet has a big role, right? Of course, it has a big role. Exercise has a big role. That helps you to boost your immune system. Exercise and having a good balanced diet, for sure.
So there's one question by Harsimrat that we are seeing bird flu cases in India in last few days. So
Mhm.
how dangerous is the bird flu transmission symptoms and death rate? So should we be scared?
So there's a bird flu cases in India. Actually, I did not follow the news, but my guess would be, I mean, are they really becoming more common and are they dying? I don't know, because I know there was a bird flu like from China earlier, right? So even for the blood flu, right? I mean, the only way is I think they through their poop, right? How they get transmitted to human, how they get exposed. Should you be worried about it? Yeah, I mean, yes, we can always, like I always give the example about, prevention is better than cure. Like, for example, I still try to go, I don't go out unless I go buy groceries and things like that. So I really don't know. Maybe I'm not the best person to answer because I didn't follow the news and what's the mechanism of those bird flu. But I would guess it's going to be very similar because you need some, there will be maybe no intermediate host. Their bird flu can directly in fact, human when you get exposed to their feces.
Sir, I have one question.
Sure.
We have seen blood plasma donation drives here in India.
Okay.
From those who have recovered, they take antibodies from them and then inject to the infected ones. So,
Yes.
compared to the vaccination process, how are these both different, or which one is more effective?
No, I mean, I will also go. The vaccination definitely was more effective, right? So the plasma therapy it has a protective. I think there's a lot of clinical trials being done, I think, in India as well, and it has a protective role. But the thing is, if you have a pandemic where I'm just giving a 50 lakh people are getting infected. You don't have that. I mean, out of those, I mean, if you can, more people can come and donate their plasma, and I think it's a great way. But it's just the logistically, right? And then you need to do a matching. You cannot just take random plasma and put it in somebody's, right? So there are a lot of testing need to be done, and how practical it is. Vaccination is something you can just go to village, door to door, and just injecting people. Vaccine is definitely a long way to. It is the best way to immunize people. So plasma therapy is good for short term, absolutely. Because when we don't have vaccine, yeah. So there are people who are in the US also they're doing the plasma therapy as well. And but the concept is same because when you put a vaccine and you actually have a much better neutralizing. You want to have a more neutralizing antibody. And same thing with plasma. When you have naturally exposed, you also have a neutralizing antibody. But my guess would be probably you do a vaccination, you probably have a much stronger, much better quality of antibody to neutralize the virus.
So we have one more question from Nidhi that if more than one vaccine become available, could taking two different vaccines boost the effectiveness?
If two, so all these vaccines are actually they are required to have two doses, right? So I got a dose, I think maybe last week. So my next dose will be the 21 or 28 days apart. So you definitely need two doses, right? Now, if you say having two vaccines means, can I take Pfizer vaccine and can I take the Moderna vaccines? And I don't think we really have the data yet, whether whether having a two dose or having a four dose can make you more immune. I don't think it is going to be that, because you already have an antibody good enough, right? And they both are targeting the same protein called spike protein. It doesn't matter whether whether you are both target is same, right? So that if the target is same, my guess is going to be, I mean, we don't have the data, but my scientific knowledge tells me that it's going to be pretty much same. It's not going to be changed much. Yes, if I'm making a vaccine against some other protein, could be. But right now, all these vaccines are pretty much targeting the spike protein, only one protein.
Okay, sir. Thank you. Sir, and then we have another question. You must be tired, I'm sure. But
Yes,
in
okay,
turmeric
keep
milk
asking.
and
I'm okay. I'm fine. If you want to ask,
I think we
go
can
ahead.
keep this as the last question. Maybe sir
yeah,
also has to leave.
I agree.
This may be
We'll take it as the
important
last
time in
question.
US, so we are
Yeah,
not.
sure. Go ahead.
We can mail rest of the questions we can mail to sir, and he can whenever he is comfortable, then he can answer those
Answer.
questions and we can share with the students.
Yes.
Okay, ma'am. So should we ask the last question?
Yeah, go ahead.
Okay. So there's a question from Shruti that turmeric milk and kadha have helped us recover from COVID, and they are somewhere a part of Ayurveda. So whom to trust, Ayurveda or allopathic medicine?
That's a big debate. I don't want to go in that country, right? So what it does is, so remember I told you there are different aspects of the COVID-19. We can have an antiviral, we can have an anti-inflammatory, we can have antibodies, we can have the vaccine. These are different arms. I don't know what's maybe the CADA or QQ, probably. My guess is, but we have the anti-inflammatory properties. If you are, don't have enough, you're really at early stage. I told you three stages: stage 1, stage 2, stage 3. And I can guarantee if you give CADA at stage 3, there's no way. I can tell you that when you are stage 3, there's no way, no matter what you give, it's because you have too much virus already in your body. Your immune system is activated. There's no way you can, put it back, right? So, like I said, prevention is better than cure. These are so really preventive type of. I can take green tea. I can take kala, cumin, milk. So these are our traditional. Ayurveda is good. I know there's nothing wrong with that. I mean, it's very powerful ways to our rishis munis have done develop those things. Yeah, this is of good. I know, but those are really good for prophylactic. When you go to the late stage, you definitely need the therapeutics. Right? Well, maybe Babaram, they must have already developed something, right?
Yes, sir. I think that's all for the day.
Yes. I think Anila.
Thank you so much for
Yeah, sure.
being so patient.
Anila, you can proceed with the official.
No, I mean, yeah, sure. So I want to encourage all of you, and also with all the women scientists. I'm very supportive of all those, putting girls, women, want to research areas. It's grateful. Don't just hang up and say, "Oh, my parent told me I need to get married and then settle down." No,
Thank you, sir.
It's very common. I have three girls, three in my own lab, and one is going back to, sir, and my parents told me to get married. Sure, of course you'll get married, no problem. But take your career, find a good husband who can support you.
Yes, sir. So first of all, thanks for giving consent at such a short notice. And when I contacted you, I was like, I used to read all your achievements at LinkedIn, so I was a little enthusiastic, and at that same time, I was a bit, I wanted कि whether मिल जाएगी permission कि नहीं sir से मांगते हैं. Let's ask for a talk from Dr. Ashok Kumar, and you were so proactive and gentle enough to give your consent, and really you have touched each and every aspect of the virus, and thank you very much, sir. On a personal note, thank you very much.
Yeah, so thanks, Sandeep. It's a pleasure to meet you online, and now we see you in person, and hopefully come to the US.
Yes,
Anybody happen to be in the US, please, just I'm telling you, please contact me. I will here.
Thank you.
Come and stay with us. I am telling you, right? And it's real. It's not a joke. I really will entertain you and feed you, and you can stay in my house.
Thank you so much, sir. It's really kind of you, sir, for these words.
Right.
I think Vandana ma'am, you can.
Thank you so much, sir, for the invitation.
No, thank you. Whatever you're doing is wonderful. That's amazing. It's incredible. You have brilliant faculty member who reach out to people, putting this together. And I think that's all. We all need to be inspired to do this sort of activity. And it's not, it doesn't cost much. I'm not traveling and you pay me, money. No. So these are the ways to all the students can engage themselves, finding the network, finding the people, right people. And I always tell all the students, and I try to help. I know sometimes we ask too much help, but I tell everyone, if your request is genuine and you're authentic, you will get it. Right?
Yes.
You have this is two things I need to have. Your request and you genuine and authentic. That's the key. You just don't go randomly on and help for this for what. You have to be very specific what you're looking for and make your network. That's the way to go in your life. Just not only science, but in anything. And that's one of the core values I develop and I share with my students and share with everyone. And just don't be shy away. Just ask people. Say the maximum. If she didn't ask me, maximum what could happen, I could have just tell them no. So what a big deal. That's it. Right? So ask those and say, can you do this? And that's it. The maximum, the worst outcome could have been for Sandeep would have been I say no. That's it. So it does not affect her. Okay, fine. I will find somebody else.
Sir, can we have a pic with you?
Oh yeah, sure. Yeah, how to do it?
So can you have the your camera is I think not visible.
Camera, sir.
Oh, it's not visible.
No, sir, we can't see you.
Oh, you the entire presentation not able to see me?
No, sir.
Wow, I think I put the camera. Oh my God, how did I did it? The coder.
Girls, you also please switch on your video.
Oh, sorry. Oh, I have two cameras.
Oh,
I thought I have my camera.
Early morning, maybe.
Wow. Maybe I was actually, but what I did is I did a wrong camera setup. I guess that's what I did. Sorry about that. I was under impression. No, everybody's seeing me, but nobody's looking at me. Okay, sure.
Thank you, sir.
Yeah. Okay, so how you can just turn off or? Yeah. So anything I can be helpful or if you need more discussion, more detailed topic, we can talk about it. All right.
So just a second. Yes, Anil, I think you can start with the vote of thanks.
Okay. Thank you, Dr. Sandeep. A very good evening to all of you. It's my privilege to have been asked to propose a vote of thanks. On the behalf of Mayor Chand Mahajan, DAV College of Women, I may like to express our sincere thanks to Principal Ma'am, Dr. Nisha Bhargava, for encouraging us to organize this enriching session and for her enthusiastic support. I further extend my hearty note of thanks to our eminent speakers, Dr. Ashok Kumar, Mr. Sandeep Pathak, and Dr. Dheeraj Kumar Nanda for accepting our invitation and gracing the seminar by enlightening us with your knowledge on various emerging strategies in fight against COVID-19 and other contagions. I am pretty sure that your precious knowledge and experiences shared by you definitely help the audiences in understanding the concept better. Thank you so much, sir. Further, we would like to grateful to Dr. Arvind Deshmukh, sir, President, Microbiologists Society of India, for collaborating with us and for providing encouragement at organizing the event. Sir, thanks for now, the special thanks to the organizing committee from the Department of Food Sciences, Dr. Vandana Sharma ma'am, Dr. Sandeep Kaur, Dr. Gurpreet Kaur, Dr. Deepika Malik for putting their efforts to make this event successful. Furthermore, let's have special recognition to all the student volunteers for providing their help in the event. And my deep sense of appreciation and thanks to all the participants who choose to be live with us and attended the webinar with great enthusiasm and made it a successful event. For this, I would like to end my speech here. Thank you all for connecting with us. Thank you, sir.
Thank you, sir. Thank you, sir, for being with us this evening. It was a pleasure. Thank you, sir.
Thank you so
Thank you
much.
so much, sir.
Thank you so much.
Okay. So I posted my email, and I think that's, you can look at, this is my department website if you want to reach out to me anytime. That's
Yes, sir, we got it. Yes,
all right.
yes sir.
Yes, sir, we
Okay,
got it.
all right.
Thank you, sir.
So
Okay,
we
bye-bye.
inching your head time and again.
Yeah, sure, yeah.
Yes. Sir.
And
Thank
just
you.
like I told you, just when anybody coming to the US, just even my email, drop me an email. I live in Detroit, and we make cars.
Yes. Sir.
Okay.
Thank you, sir.
All right.
So
Bye bye.
when you visit to India, sir, please visit our college.
Oh yes, I will definitely. Yeah, I will definitely will do. I usually, yeah. Now I have the other connections. Yeah,
Yes,
that's true.
sir.
I usually go to
Visit
PCI
and then our
I meet
college.
my professors, and that's it. So yeah.
Thank you so
Okay.
much.
All right,
Thanks.
bye-bye.
Thank you.
Chalo. Thank you.
Thank you, students, for your patience.
Students. Thank you, Sandeep, Gurpreet.